Hello everyone, and welcome to the final day of our National Cube Meet 2021. So now I would like to request Ashutosh to give us a summary of the day two of our national meet.
Yes, thanks, Karan. So, for all those who are joining on YouTube, all those who are watching this meet, let me tell you that we had this 2 days of very engaging sessions. Lot many students from across the country have presented their work on so many different model organisms. So, lot many projects being discussed have been discussed in these last 2 days, and we are looking forward to a few more projects, a lot many more projects to be discussed in today's day, the final day. So let me give you a very brief summary of what we discussed in yesterday's meet. Okay, so we began with earthworm, and these students, after having briefed us on how they culture the earthworm, they talked about all those questions that they are trying to answer in earthworm. So they said that okay, they are studying regeneration. They are doing regeneration studies in the earthworms, and they are also studying the behavior of earthworms. And then they talked about the setups that they had. So they have this something called as the uh, rapid escape response assay. Okay, where they basically submerge the earthworm in water and then in saline water, and then check for its response. Then they do a legend, okay, where they actually cut the nerve cord of the earthworm and check whether it can still escape, whether it still has that movement. And then obviously they expect that okay, whatever legend that they have created, whether this would grow back, would regenerate back. So for an earthworm which has the legend would not be able to escape out of the water, but for an earthworm that regenerates it is able to do that again. So this way they briefed us about all the regeneration studies and all these assays that they are doing. Next, we talked about Hydra. And in Hydra, though Hydra is one more organism that people use for regeneration studies, but the cubists over here were doing studies where they actually give Hydra without any supply of food for lot many days by the simple observation that when Hydra is grown with algae, the algae is incorporated within the Hydra, and the algae causes photosynthesis and it supplies food to the Hydra. So in a way, when this is done, even without any food supply, the Hydra is able to grow for many days. Okay, and very interesting uh, prospects that we discussed yesterday of how this happens, the mechanism of how that algae is taken in, and how that algae many a times there's a possibility that it becomes totally dependent on the Hydra, where if the algae now comes out after many generations, if it comes out as the Hydra, probably it would not be able to survive in its environment. So we discussed a lot on all these different possibilities. So very engaging discussion over there. Then we went ahead to discuss about the zebrafish. And in the zebrafish, certainly these students talked about how they grow the zebrafish and one the entire setup of how they maintain these fishes. And we also talked about cannibalism, where the adults, they actually eat the eggs that have been laid, and in order to save these eggs being devoured by the adults, the students talked about the setup of how they have come up with an innovative way where they could avoid these eggs being eaten. Okay, so that is what we talked about. And then we also talked about all the different areas in which the zebrafish is used, how toxicology studies are done, what is toxicology, how immune system, specifically the innate immune system, is studied using zebrafish. And yeah, so all these major questions that normally people answer using this particular model organism, and what these cubists look forward to in terms of what they would do with the zebrafish that they are currently maintaining. Yeah, so then we went ahead and we talked about the fruit fly, and this was a very long presentation with so many different aspects being taken up. So uh, the one major very interesting thing that we discussed over here is where the fruit flies normally require this medium that you are seeing on screen. Okay, is the corn meal agar medium, but then this is made by using certain chemicals that weren't available to the students during the lockdown period. And so the students very innovatively came up with their own kitchen-based medium, home-based medium called as the TRSV medium, the tomato rava, the tomato rava sugar medium, right? Vinegar medium, and which is something like cooking a dish at home, and then they are actually pouring it into these bottles, and without using any chemicals, they are actually providing all these nutrients to the fruit fly using this particular medium. So that was very interesting to learn. And so basically, the one interesting thing I would want to say about what discussions we'd had yesterday were these home lab innovations that all the students were talking about. Even with the case of the zebrafish, even in the case of the earthworm, they were talking about these solutions being made, the earthworm being submerged in water, plain water, and the earthworm being submerged in 200 millimolar of NaCl solution. And then how did the students come about, calculating 200 millimolars? How did they prepare a 200 millimolars NaCl solution at home without the help of a measuring device, where they could weigh the chemical, weigh the salt, and how they came up with innovative ways. So that was really, really interesting to learn. Again, with the fruit fly group, how they came up with designing a medium. And yes, we certainly discussed many other aspects in fruit flies as well. Then we talked about we also talked about the makerspace activity, where so many activities that are done by the students, how these could be supplemented by certain DIY, do it yourself, basically tools that could be made that would assist in sensing a few things, as in measuring certain parameters that are required in all these projects. So to take up an example, that many of these students are studying on the cardamom plant, and they are trying to study when do the leaves of this plant actually close. Okay, so during sunset, during sunrise the leaves open up, during the sunset the leaves close. And and the students are laboriously actually they are noting the angle that they make with the stem, and they're noting it every single hour. And how then all these DIY projects could help us make the job easier for the students. And not just this, we discussed many other projects. So we also talked about how we could have different kinds of sensors that could be used to assist these projects. So that was a very engaging discussion that we had, a very long discussion and an engaging discussion that we had. And then lastly, what we talked about was on nail regeneration. And this, we actually were discussing a lot on in understanding what is regeneration, what do we mean by growth, what is the difference between growth and regeneration. So the hair that grows, do we call that as regeneration? And the nail that grows, so when we call something as hair growth, at the other end we have nail regeneration. So is that also nail growth, or that is regeneration? So basically we revolved around understanding what is regeneration and what is growth. So there were so many different opinions that came up, and we were discussing this. And towards the end of this particular presentation and this discussion, we also talked about how the growth of the nail could be used as an indicator of the health of an individual. Okay, because many students were discussing how the rate of nail growth is different in females as compared to males. The rate of the nail growth differs with age. So towards the later part of the discussion, we also talked about how it becomes an indicator as to what basically an indicator of the health of an individual. So very engaging discussions, very engaging discussions on all these different projects. And in these last 2 days we had all these discussions. We are really hopeful that even today's discussions would be equally engaging and yeah, interesting. So I think that's it with the summary. Over to you, Kiran.
Yeah. So thank you so much, Ashutosh. You have told us like all the things that we had discussed yesterday, and I would say that whatever activities or experiments that Cubers are actually doing, they are inexpensive and are only made and done with readily available materials. so that this actually this core component of science curriculum, this can be implemented by everyone and even in all schools, and even with including those with have like inadequate infrastructure. So I really appreciate this thing. So now I would like to welcome Mayur Gaikwad, who will be chairing the session today. So welcome to Mayur, sir.
Hi, Kiran. I hope I'm audible. Hello.
Yes, yes,
Yeah.
you're
Yes.
a lot.
Yes. So, hi guys. I'm Mayur. I these days I teach in Elphinstone College Biotech department, and so we have lot of model systems today. We will be going through moina, snails, nematode, mosquito, ant, and pigeon. Also, we will be seeing all those videos. Something about me. Uh, as I told you, I teach in Elphinstone College in Biotech Department, Degree College. I very proudly say that I'm a product of CUBE. I've been doing CUBE activities. I've been involving in CUBE from my first, second, third year, and then also with that, I was mentoring during those days, even during my masters, and even now I'm actively involved. So whatever good or bad, it's on CUBE. Okay, yeah. So I guess I will not take much of time, and I hope Moina Group is ready for uh, for the presentation today or showing the video today. So I will request Moina Group to project their video. First of all, introduce themselves and then project their video. And I'll ask Snail Group to be ready also. Okay. Yeah. So over to you, Moina Group. Hello.
Yes, Mayur, sir. So over to Moina Group. Please introduce your. Yeah.
Yeah, yeah. Hello, all. This is Drishan Kavle from Elphinstone College. I am a second year biotech student, and yeah. So, Kiran, ma'am, you can upload the video right now. I'll introduce my other collaborators. I have Zara from Elphinstone College and Rachel Mann from CUBE Ranchi, and we have Batul ma'am from Kolkata, and a few more collaborators are yet to join. So till then, we can proceed.
Okay, so thanks, Rishan. I will now present your video.
Yeah, please play it from the start.
ये हमारे मोइनाज हैं और इनकी
हो रही है पार्टी हो रही है पार्टी 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 हो रही है और ये हमारी पार्टी पार्टी
milk drops, it turns into red in color. By adding more milk drops, we are creating low oxygen level or hypoxic condition for Moina. We hypothesize that this red color could be hemoglobin, which is also present in our blood. Our hypothesis is very similar to the Nobel Prize in Physiology or Medicine 2019, which was won by William Klein, Sir Peter Ratcliffe, and Greg Samanza. It describes how cells sense and adapt to oxygen availability. They describe the molecular mechanism behind it. Anorexia means in normal oxygen level, HIF, hypoxia inducible factor, rapidly degrades, but in hypoxia means in low oxygen level, HIF accumulate in the nucleus and binds to the specific DNA sequence in hypoxia regulated gene.
Histone acetylation, an enzyme known as HAT, histone acetyltransferase, is responsible to carry out acetylation of DNA and histone protein. HAT attaches itself to the histone protein, which in turn changes the overall charge of histone from positive to negative. The newly formed histone and HAT complex repel from the DNA strand. The repulsion causes the DNA strand to unwrap itself from the protein. The transcription factors work in action to carry out transcription on the uncoiled DNA strand. Histone deacetylation. In order to coil the uncoiled DNA, an enzyme called HDAC, histone deacetylases act on the complex. It breaks the attraction between the HAT and histone protein, causing the deacetylation of DNA strand. It coils back around histone protein into the compact closed form. Thus, HAT and HDAC enzyme act as switch for on and off of the gene.
Yeah, so it was amazing video. All of you, congratulations, Naina people. So we have huge group over here. So before you start asking questions or before you, so I will give you some time to think about the questions on the video which you have seen, which was amazing video. They have worked a lot. And if I a little bit history about Moina in Cube. So when Moina was started, when I was there in my first year, that was 2012-13. That time, one of our seniors, who was my senior in my college, he said that we work on Moina as an experiment on heartbeats. We see the effect of some copper sulfate on heartbeats. And this was the question. So I guess Ashutosh was the project head there, and Arunan sir was there, and they both said, why don't we bring some animals here? And so what happened is, so we brought from our college CHM pond, and after we brought this, and we started struggling to culturing them. We were not knowing about how to culture and how to maintain them. So after this kinds of discussion, we started culturing in water. Then the Moina died. Then we started addressing the issue. The water must be having chlorine, so we started with dechlorinated water, and thus we started improvising or the Moina group started improvising the theory, the first batch of Moina. And after that, there is very amazing thing happened in lab, Cube lab. That was school children came in, and when school children came in from Adarsh Vidyalaya and nearby school in Mumbai, and they actually started collaborating with us, with the Moina group. And one of the guy did a huge goof up, and what happened? His Moina turned red. And when his Moina turned red, that time when we used to have evening causeries, and these when people were talking about these causeries, that time he was not saying that what he.
Mayur, we lost your voice.
Hello.
We can hear you now. Yes.
Yeah, so I was saying that when one of the wonderful thing happens, that was uh, school group came into the Moina group, and they started addressing this question, and one of the guy did goof up, and his Moina turned red. And when after in causerie, when we asked continuously, then one guy said that, sir isne he was saying this to Arun sir isne दूध ज्यादा डाला, एक drop की जगह ज्यादा गिर गया. And then the question started, we started addressing question why this happens, and there was a huge discussion on that. And uh, the next milestone happened when the collaborator from Delhi came, ANDC, when they joined. A very peculiar group, a very peculiar girl. There is Kajal. I guess she is not with us today. But Kajal, when Kajal came in, she was addressing this question of this. That time we used to call it Daphne. I'm very sorry. That time we used to call it Daphnia. And when VS College, Vivekananda College came in, they started calling it the chosen Daphnia species, TCDS. And this Kajal was not happy about it. Kajal told, okay, I need to know what the species is, at least genus is Daphnia magna hai, Daphnia pulex hai, kya hai. And then she took it to Delhi, and when she started addressing this question, uh, when she started addressing the question on in Delhi, and then she asked, okay, let me find out the species. Then she collaborated with some some Pune Research Institute, and after that she once she thrown a bomb on us that it is not Daphne, it's Moina. And then if you we have all the email records of that, then people start asking her questions, lots and lots of questions on what is this, why you are telling Moina. And she then convinced, very tough it was, but she convinced us that it is Moina, and that's how we have started talking about Moina now. Yeah, so here we are now, after I guess 3 to 4 I will not say generation, but 3 to 4 batches. And that's why we can see very easy understanding of, we can understand about what is hat, what is HDAC. And this discussion has come from 2013, years after years, batches after batches, and such a wonderful people contributing inside. Okay. So Daphne has a huge history in Cube, I guess. Okay, so I will stop now, and I will open this session for question and answers. Okay, and I hope Moina group, the huge Moina group, is still around. And yeah, so anybody who is asking questions, and Saida, I guess you can take the questions from YouTube also. Okay. Yes, anybody wants to ask any question on the video?
Yes, so there is one question in the chat box which says, so if these genes are turned off in normal oxygen condition, then does it mean that hemoglobin is absent in Moina in normal condition, only produced during low oxygen levels?
So when we see that hemoglobin is not expressing in normal condition, so it doesn't mean that Moina is not having hemoglobin at that time. It means that the hemoglobin is not producing that time, uh, as there is mechanism like acetylation and deacetylation. So at that time, as this in whiteboard also, it can you just show this? Yes. So here we can see that in normaxia, means in normal condition, it ultimately degrades, but in hypoxia, uh, genes can transcribe the desired gene which is responsible for hemoglobin protein synthesis.
Yes.
So it means yes.
Yeah, I'm very sorry, Rachel, to mention. You're talking about there is something is degrading, and when you see this graph is related with that Moina question, but I guess if you talk about yes, so can you tell us in detail about, like, for example, when you talk about in normoxia, when there is no oxygen, correct, or there is very sorry, when there is proper oxygen, that proper oxygen will lead to what? That proper oxygen will lead to hemoglobin synthesis or no?
So in proper oxygen, it simply means that there is gene present in Moina, but in normal condition, it is not expressing itself. But in hypoxia, it is expressing itself. It means the gene is able to express in hypoxia.
So
But
talking about, yeah, sorry, yes.
go ahead, Saida.
So talking about hemoglobin production during normoxia or hypoxia, hemoglobin as it is gets produced during, like, in normal condition, also in hypoxia. But the thing is, during hypoxia we hypothesize that more hemoglobin gets produced. That's why the Moina is turning red. It's not that when Moina is colorless there is no hemoglobin produced. It is produced, but in less amount, like in
See,
humans. Yes.
see, do you have any experimental proof which talks about that there was a normal oxygen and there was no hemoglobin or less hemoglobin, and there was more oxygen and then there was sorry, there was less oxygen and there was more hemoglobin? Do you have any experimental results or something which you have done which proves this theory?
So yes, we have done it, but it's not like we want to do it once again because it was not done properly for the first time when we tried it.
Yeah, so
We
what you did, just tell us what you did exactly.
so so we hypothesized that there would be more genes for hemoglobin, and so for example, if there are 4 genes for hemoglobin, only 2 of them produced hemoglobin in normal condition, and rest of the 2 are blocked binding to the histone. And in hypoxic condition or in low oxygen condition, all the genes are producing hemoglobin. So with this hypothesis we tried using valproic acid, that is HDAC inhibitor, that is histone acid. Can we write it somewhere on the board? Histone deacetylase inhibitor. So something that we study in curriculum was um, histone modification, as in
histone usually binds to the DNA, right? Because it is positively charged and DNA is negatively charged, so it is coiled around histone. DNA is coiled around histone, and if the gene is if the DNA part that is bind to the histone is actually having the gene, say hemoglobin gene, the histone is to be removed from there. So there is histone acetylase which attaches acetyl group to histone, making histone negative, and now the DNA is open. Can someone draw some of these things or write some of these things on whiteboard? That would be helpful. So uh, RNA, so the DNA is free for RNA polymerase to make the protein. Whereas in normal conditions, histone is attached to the DNA, and that is how uh, like less hemoglobin is produced. So even so we did one experiment where red moina is turning back to colorless when put into normal condition. So that means, of course, when we come when we put moina back to normoxia, normal oxygen level, again hemoglobin is produced less. So there's this enzyme histone deacetylase which removes the acetyl group from histone, making it again positive. So so for this experiment we used HDAC inhibitor, and the moina continued to be red in normoxic condition. But we want, because there were some holidays in between while we were performing this experiment, so
Yeah.
this,
So
yes.
so what exactly? See, we are still not understanding what was the experiment. Like Omkar is asking specifically that how can we say that oxygen is less in particular region? And the second question he's asking that why to study hypoxia and normoxia? What is its importance? So I guess Saida, if you talk about what exactly you did with proper experiment, your experimental design, then it will be very clear for us also to understand that why you're saying all this.
So valproic acid is available in the market. So we made different concentrations of valproic acid and added in a normal culture. So we usually culture moina with 1 drop of milk as shown in the video. So and that we considered as normal culturing level, like normoxia, where the oxygen level is normal.
How do you know that there is oxygen condition is normal? What is the indicator of that?
So A, that the moina is colorless, and B, we have also tried doing DOD or DOD, that is dissolved oxygen demand. So because of which we
See,
know.
BOD, can you anyone write BOD there? BOD is, I guess, what is BOD? BOD is biological oxygen demand. Okay. So you're saying that you use that assay. So can you just tell about a little bit about that assay? Okay, Arun sir is asking that do you have some more slides to tell your whole story? If you have some slides to present, please do that, and then we can come to this discussion again. Saida, if you have some slides to present, so please do that.
So while showing the slides, you can also talk about that what you are showing. And anyone from the Moina group?
Yes, so yeah, I would like to proceed from here. So the question that we are addressing behind like this model organism is that, as Mayur sir said, that there was a goof up that was like before, I think 7 to 8 years, there was a goof up done by a school student that he added extra drops of milk in the moina culture, which led to the moina being colored after some time. So like after some days. So why? So the question here is that why did the color change took place in that moina on addition of the extra drops of milk? Now to just find so if we just normally say that yeah, that would be because of low oxygen and stuff, people won't believe us totally. So we need to design a few experiments to go about like convincing people and to have a basis kind of uh, for what we are saying. So because of which we designed the primary experiment of turning a colorless moina to a colored moina under a specific condition, wherein the control setup was, so our initial culturing setup is that in 250 milliliters of dechlorinated water, we add 1 drop of milk daily. And we observe that the moina which are there inside that culture, they remain colorless throughout their stay in that bottle, like from the time they are born till the time they die. So they remain colorless. Like, and uh, we have a similar setup. Just the variable is that we add extra drops of milk. That is 6 drops of milk in that. 6 drops of milk in 250 ml of dechlorinated water, which results in the color change in moina. That means the moina like has a like undergoes a transition of color from colorless moina to a colored one. So a colored one means it has, initially it was colorless, then it turned to pale yellow, then somewhat orangish red, and then finally red. Like not so red is different for different people. So that is why I'm just saying colored moina or a dark colored one. And yeah, so this colored moina is achieved after, so the first color change in the moina is seen after 3 days. That is the first color change that I said, pale yellow color, that is seen after uh, the 3 days after adding like these 6 drops of milk. And we are adding these 6 drops of milk on alternate days in that 250 ml of dechlorinated water culture. And we have replicates for this setup as well, like 2 replicates in all, sorry, 1 main and 2 replicates for this, in total 3.
So do you also take in consideration the number of moinas when you are planning these assays? For example, if you talk about in 1 drop, there will be, if there is less moinas and in 6 drop there is more moina, the situation will be different, correct? So do you plan any specific or is it just random?
No, we have a specific number of moina that we have. We have 10 moina initially on day 1 while we are preparing the setup.
Uh, in between, Divya is asking that is it possible for us viewers to view moina in our homes? How may we do that? So I guess Divya, you can see what Drishtant is doing for the 1 drop thing. Uh, it's just plastic cup, and in that he's adding 250 ml of water, which he has kept open for some 20 hours or something, so that chlorine evaporates. So that water he takes and add into this cup, and then he uses 1 drop of milk, and moinas are there in that. He brings moina from the previous culture or from ponds, and this is how we culture moina, I guess. Yeah, so Drishan, you can go move forward with that.
Yes, sorry, I was disconnected for a while. Yeah, so answering to Dia's question, yeah, as our Mayur sir said, we have been doing. So I would like to tell one thing very proudly that so I and my collaborators. So I'll show the map of ours. Yeah, so this is the Moina collaborators map. We have collaborators in Mumbai, we have in Kerala, we have in Jharkhand as well. Man, Rachel, Manjuel are from Jharkhand. Ram is also from Jharkhand, Ranchi, and we have Batul ma'am in Kolkata, and recently Shrudhika from Kerala and Ishita in Assam had drawn out a few water fleas from the nearby water bodies. So I proudly tell that me and my collaborators have been successfully culturing Moina in our respective home labs for like more than 1 year now. And we have been doing it very in a frugal manner without so much of sophistication, and we have been successfully maintaining the Moina till now. And some of us also have carried out the experiments that we were describing sometime before in our home labs only. So you can see that this model organism is very approachable and very easy to handle. And talking about the visualizations, Dia, so I think we have a photo of that. So Rachel from Ranchi had so visualize these Moina under the from in a jugaad way. I think Rachel would be the right person to describe their setup. Could you please describe your setup, Rachel, to visualize the Moina from a laser light?
I would like.
Yes, can you please present that picture showing the
Give me a minute. Give me a minute.
yes. So Moina is organism that can be seen by naked eyes. So if we can see some dots, but for visualizing more, like its for its identification, and so we used laser light lens. So it is a laser light is a toy, and we uh, used its lens and attached to the phone camera, and we just kept one drop of water and then Moina in it. So we found we got the picture like this, which is very much similar, which was taken by the microscope. So here the magnification used in the microscope was the 10x eyepiece and 10x objective lens, and here we used a laser light lens attached to the phone camera.
Rachel, what is the size of Moina? Arunan sir is asking.
So it is around 1 mm to 2 mm.
Okay, millimeters. That is 1/10 of a centimeter, right?
Yes.
Yeah, and how much does this setup cost? The jugaad way of visualizing Moina at home.
So we all have phone, and the laser light may cause you around 20 or 25 rupees. Yes.
Can you,
Yeah.
sorry for disturbing. Can you tell me what a laser lens is?
So what actually laser lens is? So we do have it is a kind of toy which projects the light in one beam in a straight in a straight line. So we use that.
Yeah, there's a confusion between laser light or laser lens. I think you use laser lens, right? So we have that laser light which has a lens inside. So these guys took out the lens and they used it for visualizing the Moina by keeping it in front of their phone
Yes.
camera lens.
Yes.
Yes, we have another question from Afroz Sayed, and he's asking, he got confused between Moina and Daphnia. So he's asking, how can we identify Moina?
So here in this picture, we can see that Moina and Daphnia look very similar, uh, but if we see in this picture that the major difference is the presence of antennae. So in Moina, we can see the antennae are present, which is near the mouth, and in Daphnia, there is no antennule is present. This is a beak-like structure in Daphnia.
Yeah, to me it appears like a cigarette-shaped structure, Rachel.
So we are actually running out of time. So do we have some questions from YouTube as well?
Yes, I think we should take few of them. So Afroz is asking that how can we make Moina red and where can we find Moina?
Can I answer?
Yes, yes, go ahead, Zara.
So it is very easy to make moina red. Is that we have to take few colorless moina, like approximate 5, and put it in 250 ml dechlorinated water, and add 6 drop of milk every day, or we can even add alternative day. So in a week we can get red color moinas.
And also he's asking that where can we find Moina?
So moinas are found in a pond or lake, what are ditches, as we said in the video before.
Yeah, they're basically found in freshwater bodies such as ponds, lakes, and yeah, and we can find it in aquariums as well because it is also used as a feed for the fishes. It is very economical to culture moina. So these guys use it to culture, to feed fishes as a live feed for fishes.
There's one more question from our YouTube viewer. They're saying, what is the objective and principle of this experiment?
So yes, Moina Group, what is your objective of doing so? Why do you want red Moina, and why are you studying red this Moina?
So, like we mentioned earlier, we want to study what is happening on a molecular level that is making this moina red with just increase in few drops of milk. And comparing to humans, our cheeks get red when we go at higher altitude where the oxygen level is a bit low. So is there any comparison between two, and how is actually what is happening on a molecular level that is this moina is turning red?
Okay, so we have lots of questions. So to discuss on those questions, I guess we can put all the questions on Meta Studio. So if YouTube viewers and the participants here have more questions, so you can put up in public chat. So and then we'll discuss all these questions on Meta Studio after the presentation. So over to Mayus.
Yeah.
Before that, I would like to just take 1 minute.
Yes, Diksha. Yeah.
Can I? Yeah. So we have. So I am posting a link of the thread or the topic that we have on Meta Studio. So please go through that topic. So we have been discussing all about the water freeze on metastudio.org, and it is a very great platform to just archive all our things and archive our things and have a discussion there instead of our WhatsApp groups. Yes, we can continue on WhatsApp groups, but there it doesn't get archived. Here we can archive it, or have a look at it anytime, and then it is accessible to all without even logging in. So yeah, please have a look. I have shared the link of the same on the public chat. Thank you.
Yes. So thank you, Drishtant, Saida, Rachel, and all the Moina group. There are around 15 to 20 people are collaborators in this, and they have contributed to make this video. Now I would like to call Snail group to present. So in Snail group we have Anjani, Mansi, Gayatri, Shubham, Nishmita, Nikita, Saida, Komal Singh, Man, Batul, Anamika Singh, and Arunima. So Snail group, please come forward. So and we can start your presentation. So a little bit about snails. So we have so again, a snail has been started from the first year. I was working on snail from my batch, and it has been taken from Sophia's lab. Sophia's lab, people were working on snails, and then we developed in. After that, there are many batches of people who developed it. We have developed the questions of, or it was already developed, the questions of learning and memory and molecular basis of learning and memory. So how do we learn? Because learning and memory is very fundamental process, and we kind of take it granted. So to understand that, we take this mail. I over to you, Anjani. So you can start your video and yeah, yes.
Achatina folica. Achatina folica, a model organism for studying learning and memory in snails, is also commonly known as giant African land snail. The snail gets its name from the fact that it is one of the largest in its own species, also the fact that it was predominantly found in Africa, that is, the coast of Kenya and Mozambique. It was later transported to other parts of the world by sailors who were interested in the vegetation, and the snails were also transported. Achatina folica has a light green and black stripe on its shell. The snail, when mature, reaches up to 8 inches in length, and they reach sexual maturity after 6 months of it being born. Once sexual maturity has been completed, they mate and eggs are formed. The eggs, once laid, are up to 50 to 100 in number. Once a snail breeds, it takes around 11 to 15 days for the eggs to hatch, as long as conditions are favorable. The favorable conditions are the temperature being around 20 to 30 degrees Celsius, and also the area surrounding the eggs should be damp.
For the maintenance of snails, we place the snails in a tray and fill the tray with little bit of water in order to maintain the moisture. We feed the snails with small pieces of carrots, cucumbers, cabbage and coriander. The snails are kept in a basket which is sufficiently aerated, and moisture is maintained. The lid is closed to avoid the snails from escaping. The basket is cleaned with water every day and food is replaced regularly. This is about the maintenance of snails.
This is Eric Kandel, and he studied nervous system in sea slug. His breakthrough came in 1970, and he was subsequently rewarded the Nobel Prize in 2000. So I'll be talking about Eric Kandel's experiment, and what his experiment basically was that it was based on learning and memory, wherein he made the slug to not react to its touches, or in simple words, habituated the slug, and then he resensitized it by giving a small shock to its gills, so its defensive systems are reactivated and it is resensitized. So I recreate the same experiment with my snails. So you might ask why I'm using my snails. So the reason why I'm using my snails is because the tentacles are somewhat similar to the gills that are shown in Eric Kandel's experiment, the gills of the sea slugs. And the sea slug's gills and my Achatina folica's tentacles, they both withdraw or retract on stimulus. So I'll be talking about my air candles experiment recreated with my snails. So as you can see in this, I touch one tentacle and both of them retract. Both of the tentacles retract. This is normal because if someone tries to touch your left eyes, you will naturally retract, or sorry, not retract. You'll close both of your eyes. So this is natural. And later on, when the tentacles start to come up again, it is to be noted that the tentacle that I've touched has come a little later than the tentacle that I have not touched. In this part, I touched the left tentacle which was previously touched and retracting fully, while the right one is unfazed. And in this one I touch the right tentacle, and it looks like the right tentacle does not the right tentacle contracts while the left tentacle does not contract. And now I resensitize the snail by giving it a strong stimulus so that it forgets the habituation that had been learned earlier by the snail. So you can see it from now onwards. Here I give a strong stimulus. It makes both of the tentacles retract. And now when I touch one tentacle, both of the tentacles withdraw. Reflex action. Reflex action is an action in which an organism, upon receiving stimulus, responds to it in an instantaneous manner, so that the organism can deal with it in the best way possible. In this diagram, we can see that the hand touches a hot pan, and then the receptors in the skin, that is the heat receptors, it sends a signal to the central nervous system, which then gives it an order to retract the hand so that further harm is not caused. In this hand-drawn diagram, we can see that in the first picture, the tentacle is in a relaxed state and is about to be touched by a cotton bud. After being touched, the retractor muscle contracts and the length of the tentacle decreases. This happens because the retractor muscle receives a stimulus present on the tentacle, which is stimulated by the cotton bud. The retractor muscle then relays a signal to the cerebral ganglia of the snail that then sends a signal to the retractor muscle to retract the tentacle. This is a common example of reflex action shown in snails. To err is human, and the 2 years that I've been working on snails with my colleagues, we have made a bunch of errors. The snails that we've been working on, they were being kept in cages as shown before in the video. So sometimes one goof up that we committed in the earlier days was that we did not close the cages of the boxes that we were keeping the snails in. So in that case, the snails, they used to escape from the box and run away, but they were slow. So it was easier to locate them if you recognized your error earlier. But in case you didn't catch your error earlier, they ended up in a dry area and ended up dying due to lack of water in the area. The other goof up we made was when maintaining snails in our lab, how snails often used to trail off, and there were other people also maintaining their model organisms, where they used acids. One day, one of our snails had made contact with HCl, which caused the snail to secrete abnormal amounts of mucus, and then after an hour, the snail died.
Yes. So thank you, Anjani. So we'll open here for the questions. If you have any questions, please shoot. So I guess there are some questions from internet, from YouTube. Like Afroz Said is saying that what is learning and memory, meaning of resensitization, and then he's also asking about habituation, and he has one concern that are you not hurting the snail. So you can just give a little bit introduction about these stuff. So this will be all clear in your one statement. And then Ashutosh is also trying to see that uh, was nothing answer to both these questions a part of your observation. What is the length of retracted tentacle when it restores? How far it restores? Yeah. So over to you, Anjali.
Yes, I'll handle it. I'll go through the questions on public chat and then I'll answer it one by one. So,
Yeah, we should take it one by one, I guess.
yeah. So I'll answer question first. What is the best way of maintaining these snails in home labs for a long time? So far that I've maintained snails after that is after post lockdown period. I successfully maintained my snails at a time um, we when lockdown was announced. It was in February, and I had taken these snails home as I couldn't leave them at lab; they would have died. So I could successfully maintain them for around 9 months, and but after 9 months, we the snails unfortunately died. So for maintaining them at home, what I used to do was uh, if we could go to the yes this picture. So I used to keep them in a tray like this, fill it with water. The water was up to like 1 or 2 centimeters, and after that I would place that is cucumber or cabbage leaves, and they would feed on it, and after feeding on it they used to rest, or they used to climb around, such as they would climb on the ridges of the basket of the tray, and as well as climb on the basket itself. And then after that I used to keep it back in the box after it had been um, after it had been done feeding. Yes. Arunansa is asking that why only 9 months. And Saida is here. Could you tell your how long have you been maintaining snails at your area?
So I've been maintaining snails since lockdown, and I still have snails at my place.
Okay.
So as you all must have observed that we see snails during rainy season often.
Yes.
So we kind of recreate that in our home lab, in the sense like what Anjani shared. In a tray we are keeping water, and not like little water, not that much that the snails are getting submerged in it, but enough water to provide moisture and maintain moisture, and of course we have to feed the snails. You can feed cucumber, carrot, cabbage, coriander.
So why this water is so important here, Saida?
So sir, we have observed that like in nature also, we see snails only during rainy season, and after the rainy season we often don't spot snails. This water is actually maintained, the moisture is actually maintained, which is keeping the snails active. If there is no water, then the snails will actually get inside this shell and not come out. uh, what we call as like they if I say in simple terms, they'll go in deep sleep until the condition is again having if the condition is again the same, like till the condition is normal for them, as in normal in the sense that there is moisture.
So you say that they require moisture, they require water to survive. So why
Yes.
is that? Why it is so important for them?
So they require moisture because the snail's body, it's if it doesn't have its daily dose of moisture, the body will dry up. So they'll either go into hibernation or they might end up dying.
So in natural surrounding, how does it happen? So you are saying that only in the rainy season, which are, let's say, from June to, let's say, September, only in this months they are roaming around and reproducing and eating, and after all the months they are not. What they are doing then?
So after the rainy season ends and there's not enough moisture, they go into hiding. And what I mean by when I say that they go into hiding is that they go underground and they burrow themselves, and so that their body doesn't dry up. And once they find out that not find out, but once the moisture around them is enough, they go back to they come outside of their shells.
So, Afroz Syed is having a question that we see snails in rainy season, and how are you able to replicate the same condition? So, the is as we said that we put some water in the cage in the basket where the snails are kept. As Saida said that it is not the water is such that it should not the snail should not be submerged, but there must be enough water so that when the water evaporates, there will be some moisture in the basket. So that is how we replicate the rainy season inside the small setup of where the snails live. So there is like constant some moisture inside the basket because of the water which we are adding. Yeah, gradually the amount of water decreases as time passes, and we keep adding water every day. But yeah, we
So
have to take care that the snails do not get submerged into the water. Otherwise, they can roam.
yeah, so there is one more question on the experimental design from Sarita Kumar from Delhi. She's asking that generally snails have two pairs of tentacles. Do these snails also have two pairs? And if yes, have you tried the experimental experiment on both the pairs?
Yes, as of now, we haven't we didn't simultaneously put pressure on both the tentacles. We have done it only on one tentacle.
No, two pairs. That means four tentacles. Do your snail has four tentacles?
Yes, so our snail have they have 4 tentacles, um, but we have only tried it with the upper tentacles.
Yeah, so we have more 5 minutes of this questioning. We can take some of the question. Yeah, yeah. Arunan sir is asking, how about the feeding? What do you feed? And then we can take some conceptual questions like what Afroz is asking about, what is the learning and memory, and what is your main goal of it. Yeah, over to you, Anjali. So what do you feed and stuff? Yeah.
Yes, so we feed the snails with cucumber or cabbage or tomato, and spinach also can be feeded, but it also goes according to the results that we are expecting. If we consider if we are designing an experiment in which the snails are meant to be given coriander, then we give coriander only as the food for uh, all the days, and occasionally cucumber is given two times a week.
No, it's not very clear. You are saying that it has, if the experiment demands, it has to give coriander. What do you mean by that?
So actually, once we had when last year we had designed an experiment in which we were examining the olfactory senses of the snail, and is it attractive, and we wanted to show that the snails are naturally attracted towards coriander and they eat it as their food. So we had kept it in a diet which consisted only of coriander. We used to feed the snails with coriander. So that.
Also, I'd like to add is that um, we also add once a week is that we add calcium to the we lace the snail's food with calcium so that the snail can maintain the shell's characteristics, as that is the shell is tough. So to maintain its toughness, we add calcium to the diet, and by adding calcium, what we used to do was that we used to grind chalks, and since chalks are made out of calcium carbonate, we add them to the we added them to the we laced the food with the chalk powder, so that they had a little intake of calcium, so that the shell could remain tough.
Hello.
Hello. Yeah,
So
I'll
yeah.
just move on to
So
the next question.
yeah.
um just give me a minute, just keep thinking.
So you can say about the first question that was asked about the learning and memory.
Okay, yeah. uh could uh
Yeah.
what's
So
the question? What is Okay, yeah. Okay, yeah, Mansi,
Yeah, so
continue.
the question is, what is learning and memory? He also asked what is habituation, because you are using a lot of these words like learning and memory as habituation, sensitization. So people are not, even we are not familiar with this jargon. So can you just little bit simplify for us so that we can also understand?
learning means yeah, Anjali, can I speak? So learning means getting some new kind of information. Means, for example, if you are walking in a new city, in a new street, so you are very aware of your surroundings. Means even if you are walking, you are having a look at the buildings around you, and then the people, how they are dressed, how is the infrastructure in that place, and you are very aware about the whole situation and the surrounding, and you are trying to fit, you are trying to gain all the information that you can about that place. So that is learning. You are learning a new thing, And even if this is not a chapter or a concept to be understood, but this is a very vital kind of information that is necessary for your survival. So that is what you are learning. And then memory is the retention of that information, is how well you are able to go into the same place, like you are able to recall that information. Means for example, if we go into the same place, if we like accidentally go into the same place after like for the second time, then we will be able to recall some moments about the some information about the place, like this building, okay, this building I have seen.
So,
And yeah,
Mansi, can
that
you
is
say in
memory.
context of your model system? Yeah, Mayu sir, please go ahead.
Yes, I was just going to say that can you just give an example of snail and talk, because this goes very vague. Correct. Yeah.
Okay, Mansi, should I go ahead?
Yeah, please.
Okay, so if we take it into in context with the snails, so in the video that I had that ma'am had displayed before, um, uh, the learning and memory related to snails is that it can. So I'll first go into habituation. Habituation in the video that was shown before was that it was the snail that it had learned not to respond to the uh, stimulus that I had been giving. So, so in the video that I'd shown before, is that the first time that I had touched the snail's tentacle, both of them retracted, but after a few touches, um, they had stopped retracting. So this was habituation. So in this case, the snail had learned not to snail had learned not to retract its tentacles. So this is a form of learning.
And
And
what
then
would
I'll
be
go into resensitization.
sensitization? Yeah, what would be sensitization?
Yeah. So so first of all, sensitization would be just give me a minute. Sorry. Yeah. So sensitization would be the tentacles. They yeah. So
They
sensitization
were
would be when the
responding to the
tentacles.
touch. They were responding to the slight touch. That was a sensitization,
Yeah.
right?
Yes.
And how would you
Yes.
resensitize it once it got habituated? Once it got habituated and stopped retracting, how do you resensitize it?
Okay, so in order to resensitize the tentacles, what we did was um, either we could not violently, but we could gently touch shake the snail so that it could so that it. Hello.
So you
Sorry,
gave a
was I not
harsh
audible
touch.
before?
Yeah,
Yes, we
you
Yeah,
gave
either
a
yeah.
gave
harsh
a
touch.
yeah, so we either gave a harsh touch, like in the video that I had displayed before, we gave a harsh touch to the nail, but also other means would be like if you could like gently or moderately shake the snail, it could get a sense of feeling that it is in danger. So it forgets that it had learned that not to react to my not to react to the stimulus that I had been giving. So it gets resensitized that
Yeah.
please that
But Ranjani,
react to it.
why do you have to sensitize and then again resensitize it? If you are saying that if you once touch the snail, it goes down, and if you give repeated stimulation, then it stops going down. That is the habituation, correct? And you're also claiming that it is also a part of it's also kind of a memory. It's also kind of a learning process. Then why you don't stop to that? Is not habituation is kind of a learning thing? Why don't we study that? Why do we have to give a harsh touch and then again a safe resensitization? Why we can study habituation only?
Could you repeat your question? I didn't quite get your question.
See, when you are okay, so when you are touching the snail tentacle, it goes down, correct?
Yeah,
When you repeatedly goes touching it, it doesn't
it
respond
does
to it,
not go down.
correct?
Yes.
Yeah. So this is. Do you think this is a learning process?
Yes, it's a learning process because the snail learns not to respond to the stimulus
Yes.
that I have been giving, right? Yeah.
Yes. So why we are not learning that itself? Why we have to again should give some harsh treatment to the snail unnecessarily? Because this is also a kind of a learning, correct? So why we are not studying only this first, and then we go to, let's say, going harsh pressures and giving pick up in snail and shaking it like that.
So in the first case that we display that the snail has learned, so it has memorized to not to react. So in order for us to prove that it is also it can also for more memories that so we also give a harsh touch so that it can relearn that not to respond to the to respond to the touch.
No, the Anjali, then you are deflecting from a question. What exactly is your question, Anjali? You're asking whether you are replicating your understanding, learning, and memory in the snail, correct? What is your question? If this is your question, then just habituation is enough. If you want to study specifically sensitization and then resensitization, those sort of, then you we don't have any choice to do that. But if your question is to just understand learning and memory, then this is enough, correct?
But to check, sir, for how long the memory remains. So when the harsh comes, the memory goes away. To see the strength of the stimulus and the retention of the memory.
Yeah, so what we can do is we can give this touch, we can give the soft touches which he has habituated after some interval of time, and then we have to just check till what time it remains habituation. That itself will be a time span for memory retention, correct?
Can I
Yes,
hop in
sir.
here? I
True.
was thinking that maybe if we do that, like give him the soft touch, it won't react the same way that it will to the harsh touch, because the harsh touch is kind of bringing out its innate ability to like retract its tentacles in some ways. So I think maybe that's necessary.
No, my question is why you have to do that altogether. If you can habituate a snail, isn't that enough?
Yeah, but we were trying to check that how long does that habituation stay? Like, if we do
Yes.
that,
So
does
how
the
do you do that?
habituation
So what?
still stay or not?
So how do you do that? How do you check whether the habituation stays for longer period of time? By doing the same experiment, correct?
Yeah, we can do that after a while, like the soft touch. This was another way of checking whether if we give him some different kind of stimulus, like the snail, will it
So
react the same way or not?
what is the necessary of that? What is the necessity of that? That is my question. That is
Um,
my fundamental question. What is your
okay,
question?
so maybe we were trying to see that if we increase the intensity of the stimulus, will the habituation still remain or not? Does that answer your question?
No. What is your exact question you are addressing here, Tanika? You are saying that you want to check stimulation, the strength of stimulation. This is what you are trying to say.
It's
What is
kind
your
of
question?
collective, maybe. Like if we're looking at learning and memory, we see different aspects of it, like including sensitization, desensitization, habituation, like all of it comes in a umbrella term, right?
No. You are talking too many of sensitization. So what are these? Is it? See, just tell me what is your question. What question you're chasing? What is your exact question you're trying to study in this news?
We are trying to learn, like we're trying to understand learning and memory in the snail in all its different aspects.
What do you mean by different aspects? Like, so if you're doing habituation itself is a learning, correct?
Yeah, habituation itself is a learning in some way. So we are trying to like maybe reiterate the like aspect of that learning mechanism. So that is why we are doing this, what we just said.
So, Anjali, do you
So,
have any thing to
so
say?
when we
Yeah. Yes,
actually,
Aida.
so when we actually say habituate the snail, as in the sense that the snail learns that he's not now it is not responding to the touch that is given to the tentacle. So this is the part of habituation, that okay, the snail is remembering this. The snail has learned that, maybe say, I'm kind of humanizing it, that the touch is not that intense. But this resensitization, the part where we are hitting hard so that the snail again starts responding, is because
Saida got disconnected actually. So Kaninika, Arunan sir has written over here that if that's the touch is given here, so it says that I touch here, what will happen? Kaninika, could you please demonstrate that what would happen if this touch is given?
Yeah,
sorry,
I
I got
think
this.
yeah, so answering what Kiran just said, I think the tentacle would retract if we're doing it for the first time. After a few repeated attempts, it won't retract to that extent.
So it will go down. It will retract till what level? At this level, it will go and retract when you give first touch. So we are trying to ask what is the behavior of the snail when you first touch the snail. What is the behavior of the snail? Yes.
I think it will retract
Yeah.
completely, like according to me.
Yeah, it will retract. First time when you do this first run, the tentacle retract. Correct. And this is the length of the retraction. What Arunan sir is showing. Correct.
Yeah.
Yeah. And then if you do second time, then what happens?
I'm not very sure, but maybe it'll be a bit less, or maybe it'll be the same. I'm not
This is the
very
first
aware of
time.
the number of times it needs to be done.
Yeah.
Anjani, can you help me here?
So Anjani, what will happen if you do second time?
So second time also the length of the tentacle so for the second time I touch the tentacle, they contract the same as the first time, but during the third time, instead of contracting fully, um, can I just draw it so that it could help
Yeah, if
in
you can do,
understanding?
then do it.
Yeah.
Please.
What color should I use? So instead of instead of contracting till the point that Arun answer has made, it contracts till over here. If you could, is it visible? Yes. So from now on, the tentacle, instead of retracting fully, the tentacle start retracting partially.
And what? Okay, so you can see some behavioral change after the third touch only. That in the first the behavior was very different, and now the behavior is different. Correct?
Yes.
Then
During
what
the
happens
first time we
after?
touch.
Yeah, after sometimes what happens?
Okay, yeah. So I'll just talk it through. So the first time we touch the snail retracts completely, and after the third touch, after the fourth touch, after the fourth or fifth touch, one tentacle. So for example, if I touch the right tentacle, so the first time both of the tentacles they contracted, but after the fifth touch, if I touch the right tentacle, so the left tentacle will not contract. So
See,
the
why you are talking about left tentacle? Why are not we are not concentrating on just one tentacle and just finish that first? What happens to that tentacle after fifth touch? Does it contract, retract? What happens to that? Does the behavior change?
Yes. So it does contract, but it does not contract fully. So as Arunan sir has drawn, it contracts till that part. So it does not contract fully, as in it does not go back to 1, but it rather goes back to 5.
Okay, so the difference between the first touch and let's say sixth and seventh touch, you can see that the behavior has completely changed.
Yes.
Is it not the indication of learning?
Yes, it is.
And is it not, and if let's say if you do the same experiment, like same experiment means if you touch again after, let's say after 5 questions, let's say after 5 minutes or something, what will the behavior of the tentacle after 5 minutes?
After 5 minutes, as I've seen in my home lab itself, the tentacle after 5 minutes, it still won't retract completely.
So what do you mean by that?
So that means the snail has learned not to. So it has formed a short-term memory that not to react.
No, don't tell about short. This is what why you're bringing short-term memory. Just tell us that what happens to this tentacle. It's not retracting, correct? After 5 minutes, and let's say you continue this question. Let's say you continue the same touch. Let's say after 15 minutes or 10 minutes, 10 and 15 minutes. What will be the behavior of the snail?
Okay, so as in when you say behavior, that means the contraction of the tentacle, if I'm not wrong.
Yes.
So it will not contract. The tentacle will not contract completely.
Yeah. So you are saying that once you give this treatment of continuous touching, it doesn't contract, let's say for 15 minutes. Okay. So have you seen that till what time it does the
Yes.
same behavior?
Yeah. So so after before, when I started the experiment, I also while I was updating the group, so at that point of time I had a same question had been offered to me, that what happens. So after so I kept checking. So after 2 hours, the snail has forgotten this. Sorry for using forgotten, but yes. So when I touch the tentacle again, it goes back to 1, like the drawn on as in the picture.
So what does it indicate?
So this indicates that this snail has forgotten that the habituation.
It basically means that the snail is not habituated yet to the touch.
Or you could say that it has forgotten the habituation.
Yeah, sorry to interrupt, though it is a very interesting discussion going on, because
Yes, we have to. Yeah. So what Kiran is trying to say, I guess we are shooting our time. Correct, Kiran?
yes, absolutely.
Yeah. So we can continue this discussion on online and offline, of course. If you have very pressing question, you can ask now. Till that, we can ask another group, which is our nematode group. It is again a huge group. I will ask them to at least come and be prepared and start working on that. Anshu, Kunal, Omkar, Bivas, Mandar Chauhan, then Lydia, Garima, Man, Sukanya, Abbas, Batul, Tulika, Adeline, Shivam, Sanskriti, Gayatri, Kanika, Gaurav. So there are many people in this group. So let's start processing this. Okay. Yeah, so we will talk about these questions in our groups and everywhere about snails.
So these
So
questions
nematode is
should be,
again very interesting. Yeah.
yes, Mayur sir. So these questions should be followed up,
Yeah, Mayur.
because we have not been discussing about snails, and that should be follow up. Yeah, over to Mayur.
Yeah. So let's switch our gears to nematode. And nematode, we know that it is a small worm, okay, soil worm. And again, if you talk about the history of it, again from start, in the first batch we didn't started with nematodes, but when some people from different VS college came, and then from Delhi some group came, they started working really on the snail, and they started. So it is very interesting thing that we were working with Drosophila, and we were discussing on Drosophila's olfactory assays, which you must have discussed yesterday, some olfactory senses. And the same question they ask in nematodes, because nematode has a very unique ability, or unique faida, that it has just 300 cells, 300 neurons and 1000 cells. So that's why people started asking the olfactory question in nematode. uh, and then the thing is, very interesting thing happened, because from VS there was one girl called her name was Rebecca, and Rebecca asked the same question, whether of Drosophila of native versus lab, and she found that there are lab bred cultures all around the world. Okay, the from Sydney Brenner times. And then she asked that, can we ask this lab versus native question in C. elegans, and from that this question came into existence. And till now people are working on that. And Delhi was one of the strong group who was having nematode. Then our many groups in Mumbai also, they were culturing nematodes, and Ranchi also there were culturing nematodes. So all over the country we were having these groups. Yeah, so over to you, nematode group. Anshu must be there, I guess. Anshu, Omkar.
Yes. So Anshu, now I will start your show your presentation.
Batul, Home Lab, Kolkata. Isolation of soil nematode. On 5th July 2020, started with placing soil on raw potato and raw carrot. On 9th July, could see thread-like nematodes collected at the edge of the soil. 19th July, the nematodes were observed under the microscope and identified to be osseous tubuli. They were 1 millimeter in size. They had vulva at the edge of the tail. The nematodes isolated in December had got vulva in the center, and they had a long stoma. They had a median bulb and a basal bulb. These are characters of the Rhaptididae family. They had a long pointed tail, the hermaphrodite, and the male tail. This is the internal structure of C. elegans. Two nematodes are seen communicating over here. How do they talk to each other? On 20th of December 2020, you can see numerous nematodes growing on a raw potato slice which has no soil. It was seeded with the nematodes from the previous culture. And water is added every day. From this also, other cultures were made by using a raw potato slice and milk in the center on the first day, and the nematode is picked up with a pin and transferred. This setup is of March 21. You can see it's placed with moist tissue paper below the potato slices, and milk is added at the center to attract the nematodes towards the potato slice. The nematodes have come to the milk. This was the goop up of June 2020 when in the rotting peach I saw some insect larvae and I mistook them to be nematodes.
Set up by Ishita, CUBE Chatshaala, Assam. It was to isolate and culture soil nematodes and was kept on 28 Feb 2021. She used 4 slices of potatoes along with soil sample, milk and Western tissue paper. On 6 March 2021, she saw no growth of nematodes and the growth of fungus on one of the 4 slices of potato. On 9th March 2021, there was again no signs of nematodes and there was growth of fungus along with eggs of flies on one specific potato slice. It was because the setup was not covered properly and the milk was not in the room temperature. These are the reasons why this setup was not successful in cultivating or culturing soil nematodes.
Mayu sir, are you around?
Yes, yes. I'm sorry. Yeah. So very nice, excellent video of nematodes. Thank you, everyone. Uh, so we will have question. Arunansan is asking, can we have? It will be a good idea if you present some slides which you want to discuss more on the work in focus. So if you have any those slides, you can just put it on the screen so that people can ask questions.
Actually, that is amazing that cubists have been isolating soil nematode with two using the things which are available at home. That is potato, milk. Actually, that is amazing, very amazing.
So how this raw potato now? It is, see, it is very. I guess it's very convincing that boiled potato won't is not working that much because of it attracts so many. It has so many problems. Correct. So do we have a kind of a protocol to let's say a very robust protocol to say that okay, if you follow this protocol, then you can isolate nematodes very easily. And do we have something like that? Yes.
Yes, sir. If we take a moist tissue paper and we put raw potato slices on that, and we put soil at the periphery, at the edge of the potato slice, and one should take multiple potato slices because the pinch of soil that you pick up may or may not contain nematode. So take multiple potato slices, spread it, and in the middle put a few drops of milk. On the first day only you have to put milk. After that you don't need to put milk, and cover the setup with lightly so that the insects don't come. And definitely you will get to see nematodes coming out of the soil and coming towards the milk within say 33 hours.
Okay, so let's say we got that nematode. Now, see, I guess I saw thing. You're saying that to identify nematode based on their uh, morphological characters and to isolate. So if you want to isolate these nematodes, let's say we have, of course there will be some nematodes which will be coming. Then how do you make how do you identify them? Because there will be
To
a huge amount of
identify them
species.
morphologically, we can use ordinary student compound microscope, or we can use a foldscope and observe the features because it is transparent. So you can see the features inside it with a, and you can identify parts such as the mouth part, the stoma, what is the shape of the stoma. Then you can see what is the shape of the pharynx. Where is the vulva located? It's there in the slides. If you see the next slide, I can show you. Yeah, over here we can see the vulva is in the center of the body, and there is a round embryo also near it. So these were the characteristics of C. elegans, which we are looking for. We are looking for C. elegans in the native soil. Then in the next slide, we can see other characteristics. Here we can see the stoma or the mouth is long and narrow. That is the characteristic of Reptilida family because they are bacteriovorous. They have this kind of a mouth. Below that, we can see the pharynx. Now over here, to show that it is Xenorhabditis, I had to show two bulbs. The lower bulb is very clearly seen, the basal bulb. The middle bulb is not very clear. That's why we could not confirm it to be C. elegans, because Xenorhabditis would have had one more bulb. Though it looks a bit enlarged, the bulb is not very clear, the middle one. The lower bulb is clear. The next slide. Here again is the picture trying to show the basal bulb, the stoma, and the middle bulb. But the middle bulb I couldn't show clearly. This is showing the long pointed tail, again a characteristic of Sinorhabditis and Rhabditis family. This picture is also showing the male tail. The male tail has spicules. So this is how we were trying to identify the nematode morphologically.
Yeah. So if you're saying, let's say there is a potato, and in that potato you have you saw nematodes, new nematodes from the soil coming. Then you take that nematode and transfer it into another potato. Correct? This, I guess.
Yes, sir. I showed you
Yeah.
in the video that it can be
Yeah.
picked
Yeah.
up with a
Correct.
pin and transferred. Yes.
Yeah, so the thing is, see, there is a high possibility that you also can pick up some kind of eggs with that, with the same needle.
Yes, sir. While picking up the nematodes, some amount of bacteria also transferred along with
Yeah.
it.
Yeah, there is
And
also
the
chances
thing
that
is,
nematode
when
eggs.
it was transferred, I just want to add, it was transferred from the first potato slice. That first potato slice had soil, but the next potato slice had no soil. The nematode was being maintained without any soil, just by putting 1 drop of water every day to keep the nematodes moist, like even without soil, and it remained for about more than 2 months. So we are able to culture them, grow them in the home lab.
Yeah, so that's fantastic, Batul ma'am. But my question is again the same. Suppose if you're transferring one nematode to another potato slice, correct? There are very high chances that you can pick not only some amount of bacteria from this culture, but also some kind of nematode eggs with that, sticking around the nematode, or something like that. And there is high chances that this nematode eggs, or whatever it is, it will be from a different species altogether. So how you make
Yes, sir.
sure that this
Yes, sir.
potato? Yeah.
Chances are always there that the culture which I formed is not single line culture. So what we need to do is we to take samples from the culture which we have made and observe under the microscope and make sure that every time that the sample we are taking is of the same nematode. We are not getting different nematodes. Because if eggs have been carried, then there would be 2-3 cultures growing on that potato slice.
You can also do one thing, that the nematode which is transferred in this plate, we can make the subculture of the nematodes present in this potato slice, and we can also we can make the new potato slice and transfer the nematode from here to here. So the chances of getting contaminated nematode will reduce, and we will get the pure single line culture of the nematode.
Yes, sir. Yeah, so yeah, we can of course do that. That's a good idea about to dilute the thing, so that we get single and cultures only. So do you have any? So now, as Batul ma'am is saying that she is successfully able to demonstrate and culture this nematodes with just potato and some amount of milk. So how do we go ahead? Because the objective after getting, let's say you get C. elegans, you know the morphological features. So after that, what are you planning to do about this C. elegans?
Well, it will be a very great thing to get a native strain of C. elegans, because there is no native strain of C. elegans from India, from our country. So mostly it has been found from London, from America, from the temperate countries. So in our country, we have not yet found C. elegans strain, and not even a sister strain. The phylogeny of 3, if you see, of C. elegans, there is so much distance between cetidans and the other species of Caenorhabditis that there is a difference between that would be between mouse and man. So there is so much space for research over there. We can try and find out C. elegans from our native soil.
Yes, of course. We hope all the group, all the best for getting C elegans, of course. I guess it's time that we allotted for nematodes, or we, yes. So now I guess we should move to another group. If you have any pressing question, please post now so we can ask till mosquito group comes and start their preparation. Okay, and we have again a good group of mosquito here. Hina ma'am is there, then Mugdal, then Sarita ma'am is there, then Rahil is there, then Anvesha, Vani, Tulika, Sonali, Sakhet, Hita, then Devanshu is there, then Sankara Narayanan is there. So there are many people in this group again. Yes, so can we move on to mosquito group? And by the very nice presentation, Batul ma'am. So congratulations for Hina ma'am.
Yes, absolutely. We should move to the next presentation, though it has not ended yet. The nematode discussion. The discussion will be continued in our social sites like Meta Studio and on WhatsApp and Telegram. So now I will present the video which is made by the Mosquito Group.
Warning. To ensure safety of others, make sure that after data collection from traps you may add oil or salt to kill the larva, which will prevent mosquito breeding. Short-term objective. To identify different types of mosquito and to spread awareness about them. Long-term objective. To prevent mosquito breeding and the diseases spread by them.
Mosquitoes are easy to work on. We can find them anywhere. We can find them in any water accumulated place and can identify them easily.
Home Lab 365 Special. Making and setting up of ovid traps for mosquito life cycle study.
Trap is a device which provides a suitable atmosphere for mosquitoes to lay eggs. The products required to make an ovid trap were a black vessel, 250 ml of water, 6-inch ruler, a pair of ice cream sticks, white clothes and 2 rubber bands.
Students had set up these traps both indoor and outdoor. In indoor near water accumulating places such as near refrigerator or in kitchens. In outdoor near plants and the garden area. On day 1 we observed some mosquito eggs on our trap. On day 2 we observed mosquito larvae and eggs in our trap. On day 3 more larvae were seen. On the 11th day adult mosquito was observed. Warning. To ensure safety of others, make sure that after data collection from traps you may add oil or salt to kill the larva, which will prevent mosquito breeding. How to identify different mosquito eggs. Aedes aegypti eggs are black in color. Eggs stick to the surface of the container walls like glue. They can survive drying out for up to 8 months, that is, even after 8 months the eggs can hatch larvae when they come in contact of moist surface.
Calyx mosquito eggs are simple brownish in color. They usually lay their eggs together to form a raft of from 100 to 300 eggs.
How to identify different mosquito larvae? Anopheles mosquito larvae are the easiest to identify. They don't have a siphon and stay parallel to the water surface. Aedes and Culex larvae are very similar, but looking at the siphon you'll notice that the Culex have longer and have a lighter color. Their body is also hairy compared to Aedes.
Aedes mosquito is a simple black-colored mosquito with white marks and lines on its body. Non-Aedes mosquito are yellowish or brownish-grayish in color with no white marks on its body.
Aedes aegypti mosquito has a lyre-like structure on its upper thorax region.
It is the vector of dengue and yellow fever. Aedes albopictus mosquito has a line-like structure on its upper thorax region. It is the vector of Zika virus and chikungunya. Calyx mosquito is simple yellowish mosquito which sits parallel on the surface. It is the vector of malaria and West Nile virus. Anopheles mosquito is simple brownish mosquito. It sits on an angle of 45 degrees. It is also the vector of malaria and can also transmit Japanese encephalitis.
Yeah, so it was wonderful video, everyone, mosquito group. Uh, so we will open this session for question answer. So you can put up some important slides uh, on this. So I guess what Hina ma'am and the whole mosquito group is trying to create an awareness about mosquito, and they want to tell you that if just by looking at the mosquito type you can identify which disease it can spread, and by you can also trap the mosquito, and by trapping you can understand that how do they lay larvae and stuff like that, and then you can destroy them. Okay. Yeah, so over to you. Do you have any questions? So we can start putting up that on the chat. See, I have a question on mosquito. See, when we are talking about the trapping and everything, but does it happen all of the season? Like for example, this is um, March, correct? So what I have observed personally, that during the month of May or June, the amount of mosquito which is around, it reduces drastically, at least in places where we live. So I don't know, does this behavior is everywhere, and does season affect this mosquito breeding and everything?
Shankara, will you take up? Or okay, the breeding of mosquito is mainly seen in the rainy seasons, and by increasing temperature, what effect does it have on the breeding of Aedes mosquito? I think Shankar, can you please make your inputs on your OV trap experience which you've had?
Yes, ma'am. Yeah, the orbit trap is a device which is used to collect the eggs. Like it gives a particular situations to lay eggs. And yeah, the seasonal wise, it's vary a lot. Like in the rainy season, the number of eggs observed was very large than comparatively to summer seasons.
So Shankar, with increasing temperature, what is the variation?
Yeah, so do you have any data? Do you have any data to project that you're saying that the the thing, there is difference between increase of temperature, but do you have any data to produce that?
Mayur sir, we are planning to do this study this season, this summer season. We want to do a comparative study with increasing temperature. What is the variation in egg laying of ADs is to non-ADs? So we have started with the home lab OV trap, how to make simple OV traps and try to study the life cycle. This last few months, we've been trying to, spread awareness of identification of different stages of ADs. And as CUBE Kochi started with the OV trap, we also followed it up. And children have been trying to do this trap setup, and they have been able to identify the egg, the difference between the egg of the culex and ADs, and how the mosquitoes they differ from each other. And we've been trying to tell people like how the collection of water, even in the indoor very small containers, these mosquitoes are breeding, because this OV trap were very small food containers which we made in the home lab. And they were just the Zomato food containers which we have used with small ice cream sticks covered with white paper, with white cloth, and we saw egg laying, and then we studied the life cycle. So it is still in process, and the variation of climate change is to breeding is our upcoming work which we'll be doing. And the most important thing which the mosquito team is doing is helping people, helping students to identify, because we've been talking about the dengue prevalence, we've been talking about the prevalence of dengue again and again as an epidemic, it's coming up, and how it happens. Most of the survey which CUBE Kanpur has conducted, 3 surveys in the pandemic during this COVID-19 pandemic. It was a Google form which we had filled, and we had shared it with all the other CUBE groups. So most of the people think that it is the mosquito which is causing dengue. They don't know that the mosquito is a vector, and this vector, as a vector, it is just passing on the virus from one human to the other. So this way we are talking about it, and we are trying to do small experiments to bring about, even with the, when we talk of Kiran, if you can show the context, the map, the mosquito map.
Is
How
it
we
at,
are connecting it. Yeah, it is in the PPT.
at the last? Okay, I got it.
Yeah, it is in the last, yeah. So if you see how this model organism is being connected to different streams, when we are talking about where it is laying egg, which type of mosquito is breeding where. When we are setting OB traps, we are putting it at, we try to put it in open. So I think Shankar would like to share, and there were many cubists from Kochi who told that when we are leaving it in the open, in the well-lit areas, the egg laying was slow. And if we are putting the OV traps in dark, damp places, we observe better egg laying. So all these observations, we have been collecting our data. We have been trying to share the observations also with each other, that the dark, damp place, why are the mosquitoes coming and laying egg more in the dark, damp place than in the light and very well lit and exposed areas. So this
Yeah,
was
on the
one
same thing.
important thing. Yeah.
Yeah, on the same thing, Sarita ma'am is asking a question that is there any particular reason for black COVID traps? And have you tried, or will you like to try, or if you have not, because you are saying you have not done the experiment, but have you tried different colors OV trap, or in the future something like that?
We had tried the transparent boxes also. At least in Kanpur, I have a setup of transparent boxes, green colored and black. So the best output was in black. Transparent boxes had very few egg laying, that too after 15 days. And I mean, that logic we were also not able to understand. The transparent rasgulla boxes were used. We did not observe, maybe we were not able to see those eggs. The contrast was not there. I think they must have laid eggs, but we were not able to see, because it's easier to identify the eggs against a dark background. Yes, Shankar, please take up.
Yeah, I like our observation that the black ones it provide a suitable condition for lay eggs because the dark atmosphere in the UV traps is the correct suitable conditions for the ADs, and it have a tendency to lay more eggs in a darker area than the transparent ones.
See, you are saying that, but do we have any logic behind this? That why they prefer dark, and do we have any research done, or anyone in literature, do anyone is talking about that why they only prefer dark or something like that?
Like it thinks that it's more safe, it's more safe in the blacker ones because it's create a dark and black atmosphere. Like it's yeah, it have more tendency to lay more in the blacker ones, and it's more safe to lay eggs in the blacker ones than the transparent and any other colors because black is more darker.
Okay, do you want to say because of predators maybe? I'm just guessing because of there are chances that predators may in the light they will be it will be very well spotted and they will be eaten. That's why they prefer dark background.
Yeah,
I don't
one of the
understand
reason
it.
was that. Yeah, one of the reason is somewhat it's predictor safe atmosphere as the mother ADs.
Yes, and my second question is to actually all the mosquito group, including Hina ma'am. So you are saying that now you will try to understand the effect of temperature and the seasonal effect on the egg laying of Aedes. So you can be prepared in the next year. You can create a warning that okay, this is the time when the Aedes mosquito is more. I guess this is what you are trying to say. But so, do you have any design of your experiment? How you're going to do it?
So pardon, can you please repeat the question one
No,
more time?
no, I'm asking that you are going to you're going to study the effect of the light intensity and the season in which you get more egg laying in Aedes, correct? Egg laying in mosquito. I'm just
Yes, sir.
asking, do we have any design to execute that? What will be your design to carry out this experiment? How you will do
Yeah.
it exactly?
So we have been setting number and 8 OV traps in 4 different places. Like in indoor in itself, we have been placing 2 OV traps in a place in like kitchen and dining rooms, and in outdoor we have been placing in shady and brighter areas. So we can count the number of eggs that we had observed, and we can do the variations like the number of eggs observed in the indoor and outdoor and shady and brighter area, and the temperature also varies in both these.
Okay, so do you want to study the effect of light on mosquito breeding, or you want to study the effect of the, let's say, the season, or the season on this mosquito breeding?
The long-term objective is to study the effect of climatic conditions, actually the season, which
Yeah.
we'll
So
be taking up. Yeah.
yeah. So what is your hypothesis? That this is what I wanted to ask. What is your
Uh,
design and what is your hypothesis
our
for this?
hypothesis, yeah, our hypothesis is that with decreasing temperature, if the temperature is decreasing, we are, I think Rahil wants to say. Yeah.
Do we have board here? Can we access
Yeah,
board?
I want to just continue that what ma'am was saying. Our hypothesis was that we will get more mosquitoes outside than in inside, because in outside traps we will have open air, due to which the mosquito will easily get attracted by the uh, to the trap outside than in indoor. So we will have more number of mosquito eggs in outside in the outside trap.
But what is the use of this experiment, outside and inside? Why we are doing? Hello, hello, my voice. Hello, I'm audible here.
Yes, Mayur sir. With low temperature, we hypothesize, like he had his own point that he has observed that more of his OB trap which were outdoor had more eggs rather than the ones which were kept inside the house. And but with climatic conditions, the change in temperature, like as the temperature is decreasing, we have observed the mosquito population also decreasing, and we are expecting a decrease in population of the mosquito with lowering of temperature. And as the temperatures is increasing, we are having lots of mosquito bites here, and we'll be calculating, we'll be trying to do clap trap and count the adult mosquitoes this season in April and May, and see the variation, how the population of mosquito is varying by doing mosquito counts. So for that, we planned this in this home lab. This time we are going to do not OV trap, we are going to have mosquito traps. So we'll count the number of dead mosquitoes, which type are we getting in this temperature in this month, that is April, May, and so on.
So what is your hypothesis, ma'am? Let's say this is March, correct? Just yesterday it was equinox, if I'm not wrong, and this is one day after equinox, 12 hours day, 12 hours night. So let's say it's a very good time to start. So when we are talking about an equinox, when there is 12 hours day and 12 hours night, in this condition, what is your what is your hypothesis that how many ADs you will find and how much non-ADs you will have?
From the past study, I think we've had a data of the last 2 years. We've had the data of October and November, not of the summer seasons. But this time we are going to do a study of the summer seasons also, and we expect more Aedes mosquito in April and May.
No, in October, November, how much do you? What was your average of in October and
It
November?
was, I think, yeah. I now he will tell. Actually, he was having the data.
Yeah, in October and November, and also in December, we had more number of non-Aedes mosquito as the temperature was decreasing in the northern India. We had winters beginning, so we had more number of non-Aedes mosquito than Aedes mosquito. Like I remember that we had 40 non-Aedes mosquitoes that we were able to find, and only 15 Aedes mosquito in December previous year 2020.
So in 2020, October, November, and December, you found 15 ADs and 40 non-ADs. Correct. This was your data.
This is only of December, as far as I remember. Only of December.
And what about the October and November?
In October and November, also number of non-ADs were more than ADs.
No, but is the difference same? Like 40 and 15 is, of course, there is 20 mosquitoes different. Does the difference is also same in October and November? Because it's very extremely interesting data, because we have worked through this, correct? So if we can tell that as month is going forward, October, November, December, you can see difference on the monthly basis. So this sensitive, so do we have that data at October, November, and December?
Yes, I have that data, but I'm unable to find it just now.
Okay.
I'll try
Okay.
finding it once.
Okay, so you are saying that October is more non-ADs, December is more non-ADs. What about see, December is also I guess it's if I'm not wrong, the solstice, winter solstice, where there is small days and big nights, correct? And if we move from December, the days starts getting larger. So till March it will be 12 days, 12 hours day and 12 hours night. So what do you think in March it will be? It will be more non-ADs and less ADs?
No, I think in March what we have find it till now, that we had equal numbers. We were finding both QLEX, that non-ADs and ADs in the same ratio.
Okay, so you hypothesize that in March the ADs will equal.
Can you just repeat? I didn't get the last line you said.
My voice is not correct, I guess.
Yes, you
Hello?
were audible. Yeah, you are audible, Mayur sir.
Yeah, so I'm asking what is your hypothesis? Because see, if we are going to do an experiment, we have to set some hypothesis. So like you have discovered that in around winter solstice, it is more non-ADs. Are you saying that when it is this equinox, March equinox, you're saying that ADs will be equal to non-ADs? This is what you are hypothesizing? Uh,
Yes. During March, we can hypothesize this, and during the summer months, that is April, May, June, we will find more number of Aedes mosquito, as the temperature will rise and Aedes prefer that we will have summer months.
can we draw a graph? Let's say this is the graph. I don't know, can anyone? I cannot draw because I'm on my mobile. Can anyone help me with a graph, so we can have an hypothetical graph, and let's discuss that how what is the hypothesis is we're talking about. So yes, because we can have red one color for ADs and other color for non-ADs, so we can understand, and we can have months at this x-axis. Months means we can talk about, let's say, December, that is winter solstice, which you have data of. Then we can have March, which is equinox. Then we can have June, which is summer solstice, and then we can have again equinox, which is in September. And we can study the difference of ADs versus non-ADs. December, March.
So, Mayur sir, is that fine? December 2020, March 2021,
Yes,
June
yes,
20.
yes, yes,
Okay.
yes. I guess this we can take as a some points, because these are very prominent things which happens. So yeah, so in नहीं दिसंबर यहाँ पे सितंबर आएगा ना किरण, this will be September. Yeah, this will be September, because September is next equinox.
I'll take blue color for Aedes mosquito.
Yes.
So in December 20, we found at least 15 Aedes. So it
Okay.
will end somewhere here.
Okay.
And in non-Aedes, I'll take green color. Non-Aedes mosquito, we found around 40, 45. So it will
Okay.
end somewhere here. So
Okay.
here we can see that number of non-Aedes mosquito is more in than Aedes mosquito in December 20, which is during the winter season in North Indian.
Yes. And then what do you hypothesize for March? What is your
And
hypothesis for March?
for March, I will say that I will be able to get 30, minimum 30 ADs, and also 30 non-ADs mosquito.
Okay. So in this thing, you are trying to say that it will be equal. What will happen in the next solstice, which will be the June 21, in which there will be longer days and shorter nights?
Yeah, in June I will get more number of Aedes mosquito, as the temperature will be equal for the Aedes mosquito to lay more eggs. So the breeding will be more, and non-Aedes I will get less.
Okay.
And
And
in
what about?
September, yeah, and in September, I think I may get more number of non-Aedes mosquito, that because the humidity is also increasing, as we have rainy season in the North Indian area, and the number of Culex mosquito can also be more in this, but the number of Aedes mosquito will be more than the non-Aedes.
Yeah. So if we join this line, can we join this line, this upper line?
Yeah, yeah.
So we can have a line graph. So this is your prediction for non-Aedes, correct?
No, blue color is for Aedes mosquito.
Oh, blue color is for Aedes, guys. Yeah.
Yeah, blue color is for Aedes, and the green color is for non-Aedes. I'll just draw the line. Yeah, so this is our graph, which we are expecting this year for March, June, and September.
So, what are your expectations, Sankar?
Ma'am, we are coming to that. We have not done the seasonal variations. We are only coming to that. We have collecting the eggs, and we are trying to do like the preferable area where the Aedes.
But Sankar, you can expect, na? You must be having some expectations.
Yeah, as per my view, the summer, the number of eggs are more.
Shankar, even this Rahil has also not done this part of experiment. They are just this is just their expectation. What do you think? Do you agree with this data, or you want to say that okay, it doesn't happen like that? It will be different than this. For example, Aedes mosquito will increase as the months go, and non-Aedes will have this kind of behavior. Yes, Shankar.
No,
What do you think?
No, sir. No, sir.
It's, yeah, I wanted to add one point, that for Kochi group, the temperature throughout the year will be moderate, as they are in a coastal area. So the temperature will be around 25 to 35 the whole year. So the number of Aedes mosquito will be more, because the temperature and the humidity.
So how will be the graph looking like, Shankar, of your place?
Sankar, are you drawing or do you need any help? Um, I think we should do this as soon as possible because it's already 8 p.m.
Yes, yes. So Shankar, are you there? we can just end up by having your expectation, and then we can and we can have some one or two hypotheses, and then we can go for experimenting. Correct? So Shankar, do you agree with this or not? Let's talk about ADs. Do you agree that ADs distribution will be like this in your place also?
Yes.
If
Yeah, it will be the same as like what Rahul said.
okay, and what about non-ADs?
Non-ADs, like we are only dealing with the ADs ones. Our OV traps are only dealing with the ADs ones. We are not dealing with the non-ADs.
You don't think there will be non-ADs mosquito laying eggs in your OV trap?
No, because we have been
No,
keeping the
actually you can find non-Aedes mosquito just like roaming around, so you can just count it that way. No need of doing it in your OV trap. You can just do it normally by clap trap method.
Yes, Rahul, but yeah, we have been not come yet to that.
Yeah, so even if you're not come, what do you expect? Because this is very crucial idea to be to understand, because on this basis you can create your awareness program. Okay, you can give your data to all the prominent places, and you can talk about, let's say we have an experiment, and this can very particularly say that when Aedes mosquito will be there in our place. Correct. So you are saying that even there is no temperature difference as or temperature variation as Rahil's place has, still your graph will be same. This is what you are claiming.
No, sir, but at the time of June and July, I hope my graphs is somewhat like constant, like it's just like yeah, it will be just like this. It will be an equal way, like because we are having no that much temperature variation as Rahul's place. Am I audible?
Okay, yeah. So we have two predictions of two different places, and of course, you all should ask every other or every other group. We have all the sides, like we have northeast, we have south, we have west, and we have north and east places. So we should have all the places to try this to clap trap and just check whether it is and whether we can see this kind of behavior or not. Okay, this can be a whole big citizen science program. In we can do this all over the country during this prominent dates. Correct. Yes. So I guess if we have more questions, we can do this on our domains, on our STEM games and Telegram and WhatsApp chats. And please do post more questions for this. This is very interesting subjects. Now we will proceed to the ant people. Ant people means the ant group, and we have two videos of ant. So I guess Sitha Lakshmi, Dipti, Safia, and Ashwin are there, and uh, we can project the two videos back to back, and then we can open the session for discussion. So a very good presentation from Mosquito. A round of applause to you guys. Thank you. Yes, and group.
Hello everyone, myself Shrutika from CUBE Tatmangalam, here to deliver a presentation on the model system called ants. This is a video taken by one of our collaborators. The ant in motion is a species of fire ants. What are ants? They are social insects, which means they live in large colonies or groups. Depending on the species, ant colonies can consist of millions of ants. The queen, the female workers, and the males are the three kinds of ants. The queen and males have wings, while the workers don't have wings. Ants come under the kingdom Animalia, phylum Arthropoda, class Insecta, order Hymenoptera, and family Formicidae. This is a picture of fire ant. Body parts of ant. Ant body consists of head, thorax, and abdomen. This is a picture depicting the head, thorax, and abdomen of the ant. Habitat of ants. Ants are social insects that live in structured nest communities throughout the world. The species determine their ant habitat, whether they live in underground, in mounds built at ground level, in wood structures, or in plants or trees. This is a picture of ant colony on tiles. Image of ant colony at soil with tulsi plant. Image of ant colony, that is worker ants carrying food to their colony. Members of ant colony. There are three kinds of ants in a colony: the queen, the female workers, and the males. The first one is a queen ant, that is female. The second is a male ant, and the third one is a female worker ant. This is an ant colony I found near my home in the soil habitat. You can see a lot of ants moving in the edges. Ants' behavior before rain. The ants' behavior helps to predict the rainfall of the forthcoming days. For instance, weaver ants sense oncoming storms. They become very active and move from the weaker to the stronger branches of the trees. Around 3 weeks before the arrival of monsoon, weaver ants start to build their colonies. This is an image of ants before rain. They are more active and are storing food supplies for monsoon. Ants' behavior after rain. Heavy rains will flush ants out of the wet soil. The ants search for food and safety in higher grounds. If it is dry, they will of course feel even better. Hence, they come out of their colonies and will tend to move out from their colonies. This is a video of ants after rain. They tend to move out from their colonies. How do ants communicate? They have a special type of chemical called pheromones for communication, which helps to identify their nest and its social status. This is a video relating to the movement and communication between ants. Relationship between ants and mealybugs. Mealybugs attract ants by excreting honeydew, a sticky, sweet substance that the ants feed on. Plants infested with mealybugs usually have leaves that turn yellow and wilt, and if the infestation is not eliminated, the plant may eventually die. This is a picture showing the relationship between the ant and the mealybug. And this is a video of ant-mealybug relation. Ants and their larvae. Larvae are for the most part being held near the eggs. They form piles where the larvae might accidentally eat some of the fertilized eggs. The larval stage consists of 4 stages, during which the workers feed them with liquids. They need a lot of protein to be able to grow into the body of an adult ant. This is a photo of ants with their larvae. The ants are carrying the larvae, and you can see a queen ant with wings and a male ant and worker ants. This is a video collected by another collaborator. Here you can clearly see the queen ant, the worker ants, and the male ants that are carrying the larvae. Life cycle of an ant. Ants go through metamorphosis, a complete change in body form that has 4 stages: egg, larva, pupa, and adult. Egg. The ant life begins with an egg. Fertilized eggs became female ants while unfertilized eggs became male ants. Image showing life cycle of an ant, which is unidirectional. Interesting facts about ants. There are more than 12,000 species of ants all over the world. An ant can lift 20 times its own body weight. Some queen ants can live for many years and have millions of babies. Ants don't have ears. Ants hear by feeling vibrations in the ground through their feet. When ants fight, it is usually to the death. When foraging, ants leave a pheromone trail so that they know where they have been. Queen ants have wings, which they shed when they start a new nest. Ants don't have lungs. Oxygen enters through tiny holes all over the body and carbon dioxide leaves through the same holes. When the queen of the colony dies, the colony can survive a few months. Queens are rarely replaced and the workers are not able to reproduce. How is it connected to context to curriculum? Ants are used for studying developmental biology, genetics, which is subdivided into genes, DNA, and genome-based studies, behavioral science, biochemistry, ecological interaction, divided to mutualism, and symbiotic association, morphology, entomology, divided to myrmecology, biodiversity, split to invasive species, social insects, distribution and diversity, taxonomy based on identification of species, endocrinology, research methodology, and reproduction. These are the connections we found for context to curriculum. Research questions. Can humans communicate with ants? Can ants see us? Why do ants kill queen ants? Why do ants carry dead ants? Do ants have a brain? Are ants intelligent? Why do they make tunnels? Why do they dead fast? When they bite, do they have poison? How many types of ants are here in our world? Does all types of ants have same lifespan? How do we identify the male and the female ant? Is it possible to culture ants in our home lab? Slide prepared by and voiceover Shrutika, data collector Sita Lakshmi, Shrutika, Irfana, Shaharbanu, Aishwarya and other collaborators. These are the set of collaborators and their home labs who have been working with ants. Thank you.
Yeah, so do we have one more video? Yeah.
Hello everyone.
Just a second, I'll upload it.
Hello everyone, myself Shrutika from CUBE Tatmangalam, here to
So actually, the second video was is made by Deepthi, and Deepthi is from Cube Kochi Center, and but Deepthi is not able to join us today because she's facing some network issue. So we can concentrate on the art presentation now. Thanks.
Yeah, so wonderful video, Sita Lakshmi. There is one question prompted to you that Ashutosh is asking: how do we culture ants, and if want to feed them, what do feed them? Yes, Sita Lakshmi.
The culturing of ant so difficult because they are living a community. In a colony on the basis of the quinine, when the quinine is die, the colony is destroyed. The culturing of ant is so difficult. I think that, and the feeding, that's I think that it's not possible. Mm.
See, Sita Lakshmi, I have one point that when you had lot of questions in back of your video, correct? So most of this question, I guess, if you I don't know, if you Google it, you will find it. So I'm not understanding that what exactly you are looking for into this ants. What, so what is your question if you are doing this?
Actually, we are dealing with basically communication with ants.
See, communication in an ants is a phenomena. What is your question?
Actually, is there any social behavior connecting ants and humans? That's our research questions, basically.
See what do you mean by social behavior? We all know that they gather together, they eat together, and there is a lot of social behavior there. So are you asking is there, or you are asking how do they do that?
How do they do that like that?
So so what are your plans to achieve that? How do you answer that question?
Actually, we just observe the things, and we'll try to find out.
Yeah. So Ashutosh is asking what specific social behavior you are trying to address.
Communication.
Communication. So Aruna is asking how do we culture honeybee? Why culturing ant? So if you
Actually,
think that
culture.
how
Yeah.
do you culture, then what will be your assumption that how do we culture it? There must be some
Uh,
kind of a method.
yeah. Actually, the best way is you must pick a single queen ant for them, and actually the queen ant would be newly mated one. So they'll start to form a colony.
Yeah, Hina ma'am is asking what type of communication did you observe? So you are interested in this thing.
Actually, ants move in a straight line. So how do they communicate? Actually, they don't go roughly here and there. So they actually used to go in a perfect line. So how do they communicate in the way in that sense?
Yeah, so I guess Arunam is asking about honeybee also has queen bees. So actually, I guess he's trying to relate with honeybees. So how do people culture honeybee where they have the same kind of they also have social behavior, they also have queen bees and something like that. So can we apply a similar kind of method in ants and start culturing them? Now,
We'll try, sir. We'll try to find out like that. Actually, we have started now this project now, so we must try it.
yeah, so I guess Sudhir sir is also asking that how you're going to study communication. Even Ashutosh is asking that how you're going to test this. So of course you may not have thought about how you're going to do the experiment, but now it's a very good time to address this such questions because people are asking, and we can design and kind of experiment to study whether how do they communicate. Correct?
Yes, it's like that.
So Aruna is asking, can we look for identifying queen bee? How do we do that? How do we identify, isolate, and then start doing culturing?
The identification of Cunan size of Cunan is big that compared to other female worker ant, and it have wings. Um, that's the main identification of Cunan. And how can take the Cunan in our nest? Um, that that we want to check the because we are first time. We are first time.
Cunans have actually they have at some point in their lives.
Hello. Yeah. So there are many questions on this. So this is a very interesting thing, and can we discuss this and really we can regulate and we can make this as a model system to understand this communication and kind of social behavior, because it's very interesting, because ants are everywhere and you can get ants everywhere and it can be, so if you get collaborators you can do it. So Arun sir is asking about so why not discuss with daily updates, and what we should do is we should plan for each day what we're going to do today, and we should post this on the group, and then we should follow it up, because let's say ant is everywhere and in summer season actually I guess we find more ants if I'm not wrong. So yes, Zara is asking, so larvae eats egg, right? So how will you culture them? Are you going to culture separately? So I guess Zara, they have not planned to culture it, but I guess they will plan now. So their first plan is to identify queen ant and then take them and culture it. Correct? But I
Yes,
think
sir.
it just strangely. So ants are also an insect.
Yes, answer insects, sir.
So similar to the fruit flies. Correct. Fruit flies are also an insect. Of course, it's a little bit other, but yeah. So if we can culture fruit flies, we can culture honeybees, we can culture ants also. Arun is asking, what about winged ants? Ants with wing.
Yeah, ants with wings are actually queen ants.
So, is there any way that we can identify or differentiate queen ant from other ants? Sita Lakshmi and Shruddhika?
Yes, actually, queen ants have wings at some point of their lives. So after mating and establishing in it, actually the queen ant will lose their wings. So it's like that. This is actually the way identification of queen ant.
So, Raj is asking, how does the queen ant gets the wing? Is that way to get wings not know to male ants and female worker ants?
Vishnu Priya, can you please help me?
Actually, the queen and the male have wings, whereas the workers don't have wings. And the queen and the male ants, they use their wings, and after mating, the male gets died, and the queen ant reproduce. Whereas the workers, they keep on doing all the working activities.
No, how does this queen ant get the wing? So another question man is asking in public chat, where should we look for queen ants in the colony? When is the preferable season or time? Can anyone from the ant group discuss on this?
That we want to check, ma'am, because we are observing a nest in 2 or 3 months. So That's ma'am.
Okay, so but you must be having some expectations, or you must be referring some research paper that if you want to study the communication among these ants, and now you said that in the colony there are male queen ant and worker ants. So how do they communicate? And then it is also necessary to actually differentiate them, that who are the male ant, which one is the queen ant. Okay, so another question from Zara is that are there only female workers?
No, ma'am.
Yes, ma'am. Mainly the female workers and do the work, and the male ants are mostly lazy, and the queen ant is only mainly for reproducing, making the next generation.
Okay, so other than that, how do they communicate? So what is the another? Do you have another research question that you are trying to address in this model system?
Yes, ma'am. We want to check. First, we want to check their communication, and we want to check their. Okay. Their. That's the design of this experiment, because when we feeding the worker ant, so when the first ant is come, and that time is noted, and that that's the main thing. And when we observed observe the activities of the ants in daytime and night, that's the another research question.
Okay, so Sita Lakshmi, I would like to interrupt in between. So, as you said, the first question is how do they communicate? Because, as you said, the one ant will be followed by many ants if you give some things to feed. So, this is the way of. So, how are you going to proceed like with this? How would you do that? How would you study that? How do they communicate? What would be your design of an experiment in order to see that how ants communicate?
Please help me, Shrutika and Vishnupriya.
Actually, ants communicate with each other through pheromones. These pheromones are chemical scents. So actually, the pheromones are detected at the of the ants with their antennae. They are super sensitive, and this antennae consists of.
Okay, so this is about that how they communicate. But I was also asking that what would be the design of an experiment in order to study this? That how do you like make a design? Do you have any design?
Suppose, suppose Shrutika, you want to block the communication. You want to stop the communication between the ends. What would you do?
Actually, I could decrease the pheromone strength. Actually, when we apply vinegar into the wood surfaces where ants are mostly found, uh, they won't reach there because they lose their connection.
Can I interrupt? See, while we observe the ants, we notice that they go in a straight line. In between, they stop and touch their heads. Why do they do like that? Have you observed that?
Yes, sir. Actually, it is a first step for pheromone molecules being transferred.
Okay, so you are saying that there are, so let's say if we have an ant, can we draw an ant at least? Okay, there is an ant here. So you are talking about when it communicates with another ant, it releases some pheromones. When ant releases some pheromones, is it right?
Yes, sir. It's like that.
So it releases from where? It releases from his mouth or antenna, or from where it will release?
It is actually from antenna.
Yeah.
Using their antenna.
Okay, so from antenna they release some chemical, correct?
Yes, sir. Yes.
And this is taken by another ant.
Yes, it is actually taken by another ant using the small hairs called sensilla located on their antennae.
So you are talking about this ants are releasing some chemical, okay? So
Yes,
Arvind sir is
yes,
asking
sir.
that what kind of chemical is this? Does it have any nature, or what kind of
Pheromone. So actually, pheromones are odor-based chemicals.
means what? Like alcohol, like salt, or what kind of chemical is it? Or yeah, is it an aldehyde or something?
Yeah, even whenever I have observed ants, like they are very much seen on the things which are sweet, like sugar.
See, to getting
Actually,
attract. See,
Shrutika, would you like to make the conclusion that how are you going to study the communication thing? And you said that they communicate through the pheromones.
yeah, for that
Okay.
you have to post daily from pigeon group, I guess.
I hope my screen is visible. To show the photo of an ant, I have opened this page.
So Faraz, if there is no sound, you can talk while the video is going on.
The name of this breed is European Lahore. The main quality and uniqueness of this breed is its rose color in its beak at the juncture of beak and wattle, and spreads in an arc over the eyes and across the back and wings. And the tail, it is used as food. Are some daisy pigeons. These are mainly used as domestic one. They are friendly and quite adaptable.
So that was a short video. What is your question that you are trying to address?
Pigeons are incredibly complex and intelligent animals. So they have many abilities and skills.
For us, we are not getting you properly actually. Could you please repeat once again?
Pigeons are incredibly complex and intelligent animals. They have many abilities and talent.
You're trying to study in pigeons.
I would like to study that how they communicate and important in ecological and social.
Oh, so they also communicate pigeon? Can you give an example, like how do they communicate? Like I don't know that. Actually.
There to communicate, like make a high pitch of sound.
Okay, for what reasons? Why do they need to communicate? Why are they communicating? Hina ma'am, do you want?
Like, what kind of social behavior have you observed, and how have you conducted your study? Like, is it been on random visits of the pigeon, or have you set some experiment?
Random visit.
Random visit of the pigeon. Okay, so how have you conducted your study? Was it on daily basis, or
It was on daily basis, ma'am.
so? Did you keep some bait or some food or something to attract them to come every day?
Yes, ma'am. In order to attract, we give some food.
So what type of food were you keeping? Were they seed or fruits? So this was your home lab innovation. Like, normally pigeons were visiting, or you kept things which were attracting them to come?
I give food like grits and some seeds.
So when you are this, so you have, I guess this is pigeon what you're keeping, and there are of course, if you have not thought about a very particular questions about, but there are many experiments and many stuff which people have done earlier. The famous Skinner's cognitive experiments they have done on pigeon. There is very interesting video on YouTube if you can get. The pigeons are playing table tennis and stuff like that. So there are many interesting things which we can do with pigeons. So what you can do is we can start discussing them on the groups daily, and we can come up with the what behaviors we can understand of pigeon, and can we do some cognitive experiments also, and of course the study of the ecological stuff. Okay, Farhan. So can we move forward on this?
Yes.
Okay. So Kiran, can you take over now? I guess today's presentations are over. We have discussed moina, snail, nematode, mosquito, and ant and pigeon, and people from all the part of our country have contributed and displayed all sort of very interesting projects, and it was very interesting thing. Yeah, over to you, Kiran. Thank you very much. Manish is asking learning and memory in pigeons. Yes, of course, we can do some experiments on learning and memory pigeon. So snails will get some partners, a partner organism to study learning and memory.
Thank you, Mayur sir, for chairing today's session. Thanks everyone for joining. The presentations were really nice. Of course, the discussion is to be continued on WhatsApp as well as STEM games, because there are a lot many questions that are unanswered. So we'll continue in STEM games and WhatsApp chats. Arunan sir, can you give some closing remarks?
Hello. Saida, what did you say? Sorry. Can you hear me? Hello, Saida?
Yes, sir. Yes, sir. We can hear you.
So did you say something? Asking me to do something?
Sir, can you give some closing remarks and we shall end the
Oh,
session?
actually, we are closing. I thought we'll have more discussion. Okay, so Kiran, do you have the copies of the certificates that we announced in November 2020?
Yes, sir, I have.
So
I
can we screen that, because we have not been able to send it, and
Yes.
even the original form of the certificate, I think we have not sent to others after the correction of spelling mistakes and affiliation errors. So can we just Kiran, can you just announce it, or you want me to say something about it again?
It would be nice if you could say, because
Okay.
yeah.
Okay. So if you can hear me, because I have a little problem with the microphone probably here. Can you hear me now?
Yes, we can hear you.
Okay. So normally we have this for national meets or some occasions. We identify and award participants. So 20 November meet, we had announced a huge number of awards as usual, because it has been collaborative ones, and people from different parts of the country. I think it's enormously exciting that different parts of the country people have been joining. So there has been some mistakes in the first version of the award certificates, because the spellings were not accurate, and probably the affiliation was not accurate. So with the help of Rafiq, again redone some of those things, and has sent it to Kiran, and Kiran is screening it, probably in some random order. It doesn't matter, because all the awards are equally interesting, and most of the awards are in the name of one or the other great stalwarts in science, education, and science in particular. So here I think I can see this one is Anil Sadgopal Award for Popularization of Science and Contribution to Science Outreach 2020. So this is, again, I would like to say that 2021 awards will be announced soon, and that will be based on the active participation and engagement of people in regular Chatshaala, as well as in regular updating and discussion in groups. I think that's very obvious to everybody. I think that helps everybody. And so we have teachers, we have scientists, we have students, including school students. This is Anil Sadgopal Award. I think we will have a discussion probably later on who is Anil Sadgopal, who is S.K. Mahajan, who is D.D. Kosambi, etc. And you can also do a search, and that can be a topic of discussion. So here is Anil Sadgopal Award for Popularization of Science. Is conferred on Sham T.G. Q. Palakkad, Ganesh Paski from Ranchi, Sarita Kumar. This is Chitralekha from Delhi, Sarita Kumar from CUBE A&D College Delhi, Chitralekha from Diyal Singh College, Sudhir Verma from DDU College, Komal Singh from Gwalior, Krishnendu Roy from Silchar, Assam, Nirmal from Sitamarhi, Bihar, Hina from CUBE Allen House. You can see that most of them are teachers or scientists. Hina Mudgal from Allen House Public School, Kanpur, Kankana Das from Nursing Nursing School, HS School, Silchar, Assam, Susanta Santi from Sapekhati, Assam, Vineet Kumar from Cochin College, the Cochin College, Kochi, Manju Subramaniam is from the same college, Subramanian. Shalini Lal from the Ranchi, DSPMU University, Ranchi, Sucheta Naik from MHSS Goa, Binu Mol from Natika, SN College Natika, SN College, this has not come, I think. Hemant Jeevan Magadam is from currently working at Thiruvananthapuram. Shiny is from St. Mary's, Shiny Tomashi, Sister Shiny. Is from, I hope the spelling is right, from St. Mary's Anglo-Indian School, Indian Girls High School, Fort Kochi. Manju Shine from Paul's Public School, Fort Kochi. So this is the first what Kiran has shown. I think this is from Anil Sadgopal, Anil Sadgopal Award. Anil Sadgopal, I think I will just give one line statement. Probably Anil Sadgopal has was probably one of the first major scientists from KIFR Mumbai Molecular Biology Unit. Left the molecular biology unit to start the science education program in a village in Madhya Pradesh. That is called the Hoshangabad Science Teaching Program. One of the most admirable stuff that he has done, and he is still in it and started this program called the Kishore Bharati, and the Kishore Bharati is getting revived, and many of us are part of that. So that's the so next slide, please. Okay, this is D.D. Kosambi Young Scientist Award 2020. So you can see that most of them will be much younger than probably the earlier group. That's all the categorization I can talk about. Is conferred on the following CUBE members for their exemplary contribution to collaborative undergraduate research. And D.D. Kosambi was one of those who believed in collaborative research, and he was a mathematician started again from KIFR Mumbai. He was mathematician started, but he has his contribution in population genetics is phenomenal, and he has been also a historian. So I think he is one of the very genuine intellectual scientist, science popularizer, historian. So we have here Shipra from Patna Women's College, Anjani Kumar from Elphinstone College. I think he was here today. Mansi Prasad was here today, Elphinstone College, Mumbai. Tulika Sharma, St. Xavier's College. I don't know whether she was here today, but Kaninika was here. St. Xavier's College, Mumbai. Akansha Sunil Kumar from Ratnam College. Aditya Joshi from CHM College, Ulhasnagar. Rahul Mudgal from, you just heard him, from Kanpur. Ramdeep Cube from Tejasri, Bhim Kodda from Nellore. I don't know whether people have come because I think they have exams. Farhan Ashraf, is an engineer who participates in a lot of the citizen science projects. And Prashant again is a physicist. Sri Lakshmi, Alina, Animation College, Kochi, Bhishma Biju, Nature Bhishma Biju, Nature Mechanics. This is a group, I think I was mentioning last time also. There is a group of daughters or my daughters called Nature Mechanics, and they have contributed immensely. Last time, I think today, this time we didn't see them, but they have been writing, they have been posting updates. Amrita Lakshmi from St. Mary's, same. Hanin Haris, Mohammed Hanin Haris, I hope I'm pronouncing him correctly. St. Paul's PHS Kochi, Rokia Taseen Fatima, Kirorimal College, Delhi, Muniba Shan, Kirorimal College, Delhi. So these are all, again, I mentioned about D.D. Kosambi and Scientist Award conferred on the CUBE members for their exemplary contribution to collaborative undergraduate research. We expect more and more people to participate in much more extent of collaboration. Okay, next slide, please. I think Kiran, could you just tell me that how much time that we have before we finish? I think we have some time. Is it not 9:30?
Yeah. Yes, sir. We have time.
Okay, so here is again, I think CUBE Cure. Okay, Kishore Bharati Grant Assistantship 2020 is awarded to the following CUBE members. This is some remuneration these two people have been getting from Kishore Bharati. That's the Kishore Bharati that I was talking about, which was started in 1970s. One of the first science teaching program called the Hope Hoshangabad Science Teaching Bharati, and that's still there, and that's contributing some might into CUBE programs. So Kable, I think the spelling probably is can be corrected later. And from Elphinstone College, and Lydia Matthew from St. Xavier's College. So they have been the kingpins for running this 1 year of the CUBE Chatshaala and Home Lab. In the background, I think they have been doing this, given this award Shobhati group. Okay, next slide, please. Sorry, missing. Okay. So interesting award again, Lydia is there, probably YouTube group. So Obey's award for pursuit of excellence to 2020. Obey Siddiqui, a very familiar name for Cubist. Like he was inspirations for us to start in this type of program in 2010 or even earlier. He was with us, and he was some of us had the occasion to work with him, and he was Sobaiti has started the organization leaving the biology unit that was called and started the yes, and he was instrumental in us starting this in Homi Baba Center. There somewhere, but for their exemplary, you could see them standing out. I think we don't have to talk about it much. Congratulations. Next slide, please. CUBE. Last time I think we have shown this as the first one. CUBE Award 2020. You always see that this is during. I think this was so the in Narayana College, Natika, Trishur. I think many people joined from there, and not only they joined, they have been very active and they have been instrumental in starting several new model system based studies and also extending the current existing model systems. And Acharya Narendra College has been one of the strongest centers in Delhi for CUBE ever since probably CUBE started in 2010, 2012 actually formally started. So I think Sarita is still here, and there are several people, Asta and others are very active. Several other people like Rahul and people were all there from there. So next slide, please. SK Mahajan Awards for Popularization of Scientific Pursuit. SK Mahajan was a friend and a well-wisher and a great scientist in Homi Baba, so Homi BARC Bhapa Research Center in the biology unit, and he has been also instrumental in us developing ideas like this, and this has been SK Mahajan in 2020 on the following CUBE members for their exemplary contribution to research and development in establishing model systems in collaborative undergraduate research program. You can see that this process is going now. Even today, you heard several new model systems are being developed, or the initiation has been taking place. So some of these people who have developed and fostered the model system-based research studies have been awarded this in November. So that's what we are talking about. Devastag, St. Xavier's College, I hope that he was here. He has been participating in the last few weeks. Sulekha Sudhakaran is a grandmother from Nature Mechanics again, and she has been contributing a lot and developing a huge number of local people to initiate in local home lab movement. Reggie Biju is also from the same group, another mother and a very great activist in science education. Shija from, so those two people are from Thiruvananthapuram. Yes, their group is called Nature Mechanics. That's the name given by them. Shah is a teacher from CUBE SNB HS Nedinganda. They're very silently doing. They are silently doing a lot of work. They are not very silent, but contributing a lot from. I think the mango mapping people should look at their contribution and see that they have been contributing from almost September onwards the mango map data, which I think have not been taken care of by the mango map group. So Shija is a teacher and she has a host of students there contributing to this almost silently, and they have not been coming to this Chatshaala. I think we should be persuading them to come to the Missy is there here. I think she has been here today. I think she is from. I think she was in CUBE Nehru Arts and Science College. I'm not sure whether she is still there. She has been very active even today. Sanya Panda is from CUBE Vishakhapatnam. Not very regular, but I think Vishakhapatnam we have a group there, and once in a while that we are getting people organized there. And Rajini Thalam is from college Nellore, Andhra Pradesh. You must have seen a group of people working on the zebrafish that is motivated by Sri Ranjini Thalam. Mansoor Javaid from the other end of India, that is from CUBE Srinagar. I think he has given the record finding of mango flowering in March, the first half of March, and he has been an active coordinator of Jammu and Kashmir CUBE groups. S. Rajkumar is from Puducherry, a small group but reasonably active for mango mapping and other six and science group. Sumita Kachwaha from Jaipur University, CUBE University. They have been off and on active. I think some of them, one of them probably must have, Sobana Datta, one of them must have left the university, but I think there is a small group that is working there. The next slide, please. So we have another very interesting, to me, it's very sort of personal type of thing, because Veronica Rodriguez has been one of my very close friends, and I worked with her in KFR, and Veronica Rodriguez unfortunately passed away, and her contribution in fruit fly studies, developmental biology, and neuroscience studies has been immense, and she has been instrumental in building up several model systems from the very scratch when she was with us. So some of the things that we are still continuing are from inspired by her. Of course, Drosophila, you can't forget about it, both contributed by Veronica Rodriguez and Obay Siddiqui. So Arunima, I think Arunima's surname or initials that we don't have here, I think probably she could add it and we can change it. Arunima's CUBE, so this is for conferred on the following members. So this is for the perseverance, and Veronica is no outsider to sense of perseverance when she was alive. So Arunima is from Kozhikode. She was earlier, I think, but SN College Natika. Ashwati Suresh from Natika, Siddhi PP from Natika, SN College Natika, Lakshmi PJ from SN College Natika. I think there is a big, strong group who has been continuously participating in Chatshaala as well as for in updating. And Abhijit Vinod from St. Peter's College, Collingerry. I think he has been having exams, and still he was participating once in a while. I don't know whether he's here today. He has immense contribution to developing and maintaining model systems, including Hydra and several others from the local area, including, I think, several model systems. Shraddha from CUBE Jinjinwala College, helping a lot and persevering in not only continuing with our cardamine studies, but also in other studies like Philanthus plants, and she has been a regular contributor. Nasish, I think everybody is very familiar. Nasish's contribution to the home lab studies in cardamine seed germination and its extension to floral method using kanamycin, as I think we heard day before yesterday, I think. She is from Institute of Science. She finished from Institute of Science in Nasish. And Vaibhavi was one of the beginners in at home studies. Yash, again from Institute of Science, of course. Vaibhavi is from Elphinstone College. Yash is from Institute of Science. Yash also completed his studies in masters in Institute of Science, and I guess that I heard that he is teaching in a college in Mumbai. Ritik Bhaviskar, he is here, very, very active, extremely active. So for perseverance in the pursuit of science, we wish them all the success in furthering their contribution to the CUBE initiative. Any more slides?
Yes, sir. We have two more.
I'm sorry. I just missed out. Yes, we have fantastic awards like K.S. Krishnan Student Mentor Award. This is students who are recognized as mentors. K.S. Krishnan is another wonderful scientist, that great human being who was in again in KFR, about the unit first and the department of biological sciences, and later shifted to along with Veronica Rodriguez as well as Obay Siddiqui to NCBS Bangalore, and he is contribution to inspiration to start the programs like what we are doing is immense. And K.S. Krishnan Student Mentor Award 2020 is conferred on the following CUBE members for their exemplary contribution of mentoring new members. And you can see that several other people have now been contributing to mentoring, and we wanted everybody to be not only doing work but also mentoring others. So this Deepika Iyengar, I am not very sure whether she is here, from Bangalore. Rachel Trekkie, she has been here. I think she is from Ranchi, DSPMU. Manmazi Bek, again from Ranchi. Anjual Jojo. I think they have been very, very prominent in regular engagement of discussion, and also continuing their program in there, as well as collaborating with several others, including the Kolkata group has been actually supported very much by the Ranchi group regularly, and I think Batul will agree to that. Omkar is here today with us, and Ratnam College group has been initiated the C. elegans studies, which I think we have heard today, part of it. So Omkar has been an immense contributing in mentoring people. Nidhi Kapileshwari, I am not very sure whether Nidhi is here. She is from Goa. When she was a school student, I think she has started mentoring others, and her right now she is an engineering student. Shalu Sinha is from Patna Women's College, Patna. Her contribution is immense, because I think the Patna group is maintained because of Shalu, and right now I think she is one of the active members there. Anshu Kadam, you just heard her. I think she was here. Again, a C. elegans group person. Garima Kalakoti, yesterday I saw her, but I couldn't see whether she is here, but probably she'll be in the YouTube group. Garima has been a sheet hanger in A&D College group. Komal Kotra again has some CUBE A&D College, Delhi. One of the strong centers in Delhi. Pulkit Singh is from A&D College. Kunal Kadam from Ratnam College. Ameya Pawar, we have not been seeing him of late, but I think his contribution is immense. Anjali Chavan is from Chennai. Chennai group has been a little slow, but I think there are two people. One is Anjali, who has been still with us. I think they Anjali and a few other people are reviving the Chennai group. Abhishek Unnikrishnan is from Kolancheri. I think along with Abhijit, I think Abhishek has been a sheet hanger in maintaining the Kerala group, especially the Kolancheri group. Sujit is from Calicut University, a postgraduate student. I'm not very sure. I didn't see him today and didn't hear him today. Otherwise, he's a vocal mentor. Rani from Ranchi. She, along with her teacher, has been the initiators of genetic engineering through floral diff method, which Nasish has helped them to move on during the lockdown period. Manisha Rani, I think she presented her work yesterday, I think, or day before, I think yesterday. Kanishka Parashar has been in, I think, the Gyalsing College and shifted to Jamia Millia Islamia, and she is still a part of the mentorship program. Oindril Datta has not been seen very regularly, but once in a while we hear him. And he is the first person probably to tell in West Bengal that in his part of West Bengal, fruit flies are not there. And then I think he established, he discussed about it and celebrated the goof up, and he found out the fruit flies in his area. Also, Krishna is has been contributing immensely on fruit butterfly studies. Of late, we are not hearing her updates, but I think she has been mentoring. And today, I think we heard some part of it yesterday. Therefore, Krishna is from SN College, Natika, Kerala. Again, Krishna's initials, we don't have it here, so probably I think she can send it to us along with Arunima's. I think then we can correct it and reprint this sheet. I think two more are there. Kiran?
Only one.
Okay.
Yeah.
So this is CUBE Pure Kishore Bharati STEM Leadership Awards 2020. I think Kiran has screened these certificates and kept this as the last one. Kiran Yadav's name is coming here. So CUBE Kishore Bharati STEM Leadership. STEM is Science, Technology, Engineering, Mathematics Leadership Awards 2020. You can see that I think most of these people you are seeing in front as well as behind, and almost the kingpin who are running the show in spite of the fact that they are very busy with other things. Kiran has been, of course, totally devoted to this, and when she went during the lockdown period, I think from there she established Moradabad, she established the home labs, and has been continuing the home lab program. But this award, of course, has been given during the November 2020. Ashutosh Mulya, you heard him. I think he has been with us, and yesterday he was chairing the session. Mayur Gaikwad was chairing the session today. Rafiq, again, you hear him once in a while, but he is behind the scene doing all these things, including getting this thing organized, this certificates organized, and the list made, etc., etc. Jai Kishan Advani was the first chairperson, I think, first day's chairperson. Batul Paipola is from Kolkata. You heard her now, just now, a few minutes ago. Rahul Kumar, I don't know why you didn't see him today. I did, because if he's here, he would be making a lot of comments. Rahul was again a sheet anchor from AND College. Now he is in here for molecular biology. Sorry, I didn't use molecular biology, Department of Biological Sciences. He is finishing his master's program probably, or maybe continuing with his PhD. So these people are given the award, CUBE Cure Kishore Bharati STEM Leadership Award. Now, I don't think that anybody can doubt about their leadership. You haven't seen this in the last 3 days, except I think Rahul has been coming regularly for the Chatshaala, but today I didn't see him. I don't know whether he was there yesterday. But otherwise, you can see all of them are very, very popular people here with us. So since at this the last slide, and I lost count of this. I think somewhere around 100 awards have been given. I think last because as the number of people participants are there more and more, I think we have also got a number of awards. So I think this is a wonderful time that we had. These 3 days is not a simple national meet, but this is also the celebration of home lab movement. It's not just a home lab, but if you look at it in literature, probably you may not see the idea of home lab movement anywhere. Something similar probably you may get it in some established labs, but the home lab movement will be something probably you may not have heard about it, we may not have even anticipated it. I think probably it is because of the lockdown that we have been able to consolidate that concept much better, though our model systems were simple enough to be maintained and address the questions in home labs. So I think I was just the last point I was trying to say is that this is a celebration of 365 days or more than that of continuous without any break program, not only from 5:30 to 9:30, but the rest of the time people were working throughout the day and night. We have seen day in and day night the data has been coming in. So I think it's a wonderful thing. I personally feel that I have never come across anything like this in my reading of literature on science education program, including the Hoshangabad science teaching program, etc. Thank you very much. Kiran, over to you. If anybody else wanted to give a quick support, I saw GN here, Arjuna. Yes. Arjuna, I would request you to make some comments on this, and I think you have something to say about how to make it more durable.
I think all that has been said already. I think it's been really wonderful, fascinating, and trend setting. Not only for a program in India, a program like this arguably has never happened anywhere in the history of the world. So all of you are heroes of this revolutionary science education experiment. I think each one of us now know that come what may, our investigations will go on. And that has been demonstrated by the passion, your time. And one of the greatest thing that happened is that we seized the opportunity during the time when the country went on lockdown. We opened up the home labs. So we locked up the labs in the colleges, but then we opened up the home labs. I think this is a fascinating innovation. People keep talking about lack of innovation in higher education in India, but as you can see here, that a lot of innovations and inventions happened, and all of you are very creative. So the problem, as I see in India, is not that we are not creative, the students are not creative or things like that, but it's just that we are not meeting up the expectations. We are getting bored by the run-of-the-mill stuff that happens in schools and colleges. Once you get out of it and start looking for opportunities, and I see, for example, coming out with local model systems, I think is one of the greatest achievements of the CUBE project. Whether it is Philanthus or Moina as a local thing. And we are still looking for Daphnia in India, by the way. So this is going to be a big surprise when we come to know that there is no Daphnia in India. I mean, in the sense of wild varieties. Of course, it may exist in the labs, but we still have to find out. So this contest is still on. If any of you anywhere in the country find Daphnia in their ponds, please inform us. And using both Philanthus and Cardamine as model systems for various studies in plants is another great invention that took place. And alternative for Drosophila media. I think what we should actually do is that we should push it to the labs now and find out how TRST medium can actually be used in the laboratories across the world. Why not? Why should we use agar agar when upma rava is available all over the country? Of course, people may crib saying that it may not be standard because the contents may change from time to time. But then every experiment doesn't need that kind of standardization. So, and particularly not so much at the time when we're actually learning how to do experiments. And I think all of you must have noted that yesterday was equinox. And we are celebrating the CUBE meet exactly at the time when the planet is also very happily celebrating the onset of spring. All the people who are observing, making observations, whether it is mango mapping or not, people have already heard the koel coos all over the place for the last 1 week. And all this is because we have been sensitized by this moment to do observations, and not only do observations, to record them and share them with everybody and discuss them. The greatest achievement, I think, also of the CUBE is to inspire us to conceptualize this idea of Chatshaala. So this is even this innovation of Chatshaala itself is something that evolved because of perseverance of all of you. I mean, what else to say? You guys inspire all of us. We are just waiting for many more people to get inspired from what we are doing here, particularly those who are sitting in the universities and colleges and schools. So we have to make them notice what we are doing. And just as we are enjoying, we want the entire country to enjoy as well. And as you can see, we are not just restricting ourselves to schools and colleges. Some of the awards that were given today actually show us that parents and grandparents are also involved in this moment. And that is, I think, says a lot about it. We are actually creating a living academy. So inviting all of you for the future of education, and we should call it the Living Academy. And Home Labs is on the way of doing the Living Academy. And in any case, this is a greatest inspiration. And we inspire each other, but we are also inspiring the whole world. Again, let me tell you, an experiment of this kind has never happened in the history of science education anywhere in the world. You are the history makers. Thank you, and all the best.
Thanks, Professor Nagarjuna and Dr. Arunan. And thanks a lot for giving us this platform where actually cubists can explore and to actually increase their curiosity about the nature, about the surrounding. So thanks a lot for giving us this opportunity and giving us this CUBE project where we can explore more. So now I would request Mayur sir to say something. Is he around? No, actually. Okay, so I think we should go for the group photo. I would like to I want everyone to open their webcam so that we can take screenshots.
Yeah, before that, I would like to make a few comments.
Yeah, sure. Go ahead. Go ahead.
Yeah, so the thing that I'm going to talk about, it's a quite like interesting and very like reminiscent thing that so it's about the history of the Chatshaala that we have been doing from. So today is the 367th day. I hope I'm audible to all.
Yes, you are audible, Drishtan.
Yeah. So I remember precisely on 18th of we had our first like informal conversation. So GN sir was there from his office, and Arun sir was there from his place, and we all were there in our home, so, not home, the CUBE lab, and yeah, we had an informal conversation, and from there on, the next day, the Chatshaala started. So I wondered that if this Chatshaala wasn't like shouldn't exist only, then we would have missed out on the TRSV medium, the mobile microscopy things, and many interesting innovations that have happened during this course of time. I personally have learned a lot and have been continuing learning, and also would have been introduced to many more like new things which they didn't knew earlier, but now are a like part of it and actively contributing to it. And yeah, it's been a great roller coaster ride, if we say.
Absolutely. Thanks, Vishan. It's always amazing to hear about the history of CUBE. And rather more amazing if your name is there.
So others have to join. Can everybody join, Kiran? What is the situation? Or do we have to?
Okay, so we are 43 people, so two more can fit in the frame. Other than we can take the next slot.
Okay.
I would request two more people to open their webcam. Okay, thanks, Zara. Yeah. Yes. I think we should also smile a little bit. Thank you, thank you. Okay, so now I will switch off my webcam and request to others that who have not switched on. Okay, so Saida, okay, Shita has. GN, yes, Mayur, you are audible now.
Oh, so, okay. Yeah, I was disconnected for sorry.
GN, are you around? Okay.
Can have some more people, no?
There are more, yeah. I will try to call out the name. Sida Lakshmi,
Yeah, I think that's good. Yeah.
Rafiq is there. Preeti, Chitralekha ma'am. Omkar, please open your webcam. Okay. Isha is also around. Isha and Sudhir Verma, Dr. Sudhir Verma. Ayush. Yeah. GN, are you around?
Yeah, he's around. I can see him somewhere.
Okay, but we can't see him. So yes, I have taken screenshots. Thanks a lot, everyone. Thank you so much.
So we completed everybody.
Was not there. Sita Lakshmi, okay.
No, Kiran, I was there, I guess, in the first frame.
Okay, okay. Thanks, thanks.
Yeah.
Yeah, I guess yes, we have.
If anybody missed out, they can have a special.
Send us your photo to us.
Okay, so great.
So that was actually amazing 3 days that we have. And I guess there are many more things to be explored. And CUBE is never stopped, and still they will not be going to stop. It will be continued. Thanks a lot, everyone, for joining us.
Yeah, we'll be starting from tonight.
Yeah, tonight. And I will request that those questions that were not being discussed, so we'll discuss those questions on our WhatsApp groups, Meta Studio and Telegram. Yeah, over to Mayur, sir.
Yeah, I guess it was very interesting today, and I was not there yesterday, but I was there half day of the first day, so it was extremely interesting, and I hope to do this every day. So whatever we have not completed, let's say today, like Moina, we have not discussed, this we have not discussed. So we give that more time because we know that in half an hour or 40 minutes it's not possible to discuss every aspects. So we have these days to discuss. So I'm seeing forward to come at least 4 to 5 days a week and discuss everything with you all. And thank you everyone. Yeah.
And
Kiran, it was
last,
very nice of you. It was, you did a fantastic job of everything organizing this. So congratulations to you also.
yeah. So thank you so much, Mayur, sir. So last time also GN said when we had last meet, so he said that don't wait for another meet to announce. You can make videos anytime. So I would request every cubist if they want to make any video, so make it or put it on STEM games. Yeah, and I would like to thanks Mayur sir, Jai Kishan, and Ashutosh for chairing this event. And yes, yeah.
Yes, and Kiran for managing the whole show. Thank you very much, Kiran.
Yeah, thanks to you, sir. Okay, okay. Let's meet then at. Let's follow up the discussion in CUBE WhatsApp groups and on Meta Studio. Yeah. So bye everyone.
Bye.
Yeah. Thanks a lot, everyone.